Kindly request all delegates and participants to take their seats inside the auditorium, as we'll be starting this morning session very shortly. Thank you for your cooperation.
Check.
May I request all to please switch your cellular devices to silence, and for those joining online to please switch your audio settings to mute. A very good morning to all our distinguished guests, esteemed dignitaries, faculty members, industry partners, and dear colleagues. I, Iba,
and Rohit Kashwaha
warmly welcome all of you on behalf of the BRIC THSTI family to celebrate the 17th Foundation Day of BRIC THSTI. Today marks a moment of immense pride as we not only commemorate our institution's journey of excellence in translational research and its growing impact on advancing healthcare innovation, but also reflect, connect, and look ahead together towards building stronger partnerships to shape the future of healthcare system.
To begin with, I invite our Associate Professor, Dr. Pallavi Kshetrapal, THSTI, to kindly deliver the welcome address.
Good morning, everyone. On behalf of BRIC THSTI family, I, Pallavi Kshetrapal, am honored to welcome you all on the 17th Foundation Day. Established in 2009 with the support of Department of Biotechnology, BRIC THSTI was envisioned by Professor M.K. Bhan to bridge the gap between laboratory discoveries and clinical applications. Since its inception, we can proudly state that the Institute has remained steadfast in its mission to advance translational research that improves public health and informs evidence-based policy. A very warm welcome to all the distinguished guests, eminent scientists, researchers, innovators, policymakers, representatives from funding bodies, colleagues from RCB, THSTI. We are delighted to have you with us today and sincerely appreciate your presence at this event. A special greeting to Professor Roger Wu, Vice Dean of Research, University, Singapore. Your presence adds immense pleasure to this occasion, and we are delighted to have you to join us for this 17th Foundation Day. With no further ado, I welcome all of you to a journey of discovery and application at BRIC THSTI, and hope you enjoyed this celebration of science, collaboration, and impact. Thank you.
Thank you so much, ma'am. Next, we have the scientific colloquium where 5 PhD scholars will present their research. Each presenter will get a total of 15 minutes, 10 minutes to present their research, followed by 5 minutes for question and answers. Please note that presenters that a bell will ring right after 8 minutes, after which you need to conclude the presentation within next 2 minutes. I first invite Mr. Linus Augustin to the stage.
So, can I start? Yes. So, good morning all. I'm Linus Augustin, a PhD scholar under the supervision of Dr. Nishit Agarwal. I'm here to discuss my work progress on my PhD topic entitled as Understanding the Regulation of Protein Homeostasis in Mycobacterium tuberculosis. So, before I discuss my results, I would like to emphasize on the need of us doing research on tuberculosis. Tuberculosis is one of the top 10 deadly disease. And as per the recent WHO report, it is estimated that 1.7 billion people have been infected from this disease, and with a mortality rate of 1.25 million deaths. When it comes to our country, India, we contribute 27% of the total TB burden, and the infection rate is about 26 lakhs per year, and with a mortality rate of 3.2 lakhs. So, and which clearly indicates that the current drug regimen is not sufficient enough to eradicate this pathogen. So there is a need of new novel therapies. So why TB wins? TB is an ancient disease. It has been reported in mummies dating 2000 BC, and it has survived through generations and decades of antimicrobials till now. This heightened persistence of this pathogen is due to its various biological features. One is its thick waxy coat, making it almost impenetrable to drugs, and ability to evade immune system, ability to change its metabolic state from replicating to non-replicating state, and ability to withstand stress. All these features are possible due to its tight regulation of protein homeostasis. So what is protein homeostasis? Protein homeostasis in general is the maintenance of healthy protein via cell, which could be a pathogen or a cell. So, and it is majorly done by 3 major steps. One is protein synthesis, then followed by protein folding and degradation aspect. So through my work, I am focused on protein synthesis and degradation aspect. So in this talk, I'll be particularly focusing on protein synthesis, particularly on translation termination. So in the case of Mycobacterium tuberculosis, initiation and elongation is well characterized, but not the termination. How the termination takes place, what all are the proteins associated, nothing is characterized. So our lab is focused on understanding how the translation is taking place. So through my PhD work, there are 2 questions which I am addressing. One is how the proteins are getting released and how the proteins are getting degraded. So in this talk, my focus is on translation termination. So what is my hypothesis? Very simple hypothesis. During active translation, when the ribosome is actively translating the polypeptide, it encounters a stop codon. So there are no tRNAs specific to these stop codons. So at this point, proteins which mimic the tRNA structures comes in and release the polypeptide. And these proteins are called release factors, and we are trying to characterize these proteins in MTB. So from the literature, it is clear that in prokaryotes, there are 3 release factors. PRF-A, which has the ability to recognize UAA and UAG. PRF-B, which has the ability to recognize UGA and UAA. And there is another release factor which aids in the dissociation of these proteins. So how to consider any protein as a release factor? So it requires 2 major critical motifs. One is the GGQ motif, which is associated with the peptidyl hydrolysis, and the another one is the PXT motif, which is associated with stop codon recognition in the case of PRF-A, SPF motif in the case of PRF-B. And there is a role of PTM, post-translational modification of the GGQ motif by methylation, which is done by a gene called HEMK. So in MTB, there are 2 genes which we are focused on, RV1299 and RV3105C. These genes are predicted to behave like a release factor. And from the sequence analysis, we expect these particular sequences to behave as these motifs. So what is my research questions? We are trying to address whether these are indeed the release factors or not, whether the motifs which we assigned are actually the motifs which is associated with translation termination. Do methylation has a role in translation termination? So my objectives are based on this, to characterize these proteins, understand the mechanism associated with the translation termination in Mycobacterium, and also what happens when we deplete, how it affects the pathophysiology. So what is my work done? How I did it? So initially we try to characterize these proteins, whether they are release factor. Then we try to understand the in vivo essentiality of these proteins in Mycobacterium, followed by probing the specificity of these proteins in recognizing the stop codons. So to validate whether these proteins are indeed release factor or not, we took advantage of a protein called NanoLuc. NanoLuc has a catalytic domain towards extreme C terminus. So imagine a situation when this protein is actively synthesized and getting stuck in the ribosome due to the absence of a release factor. It means that the catalytic domain is within the ribosome, due to which it will not be able to cleave the substrate. So there will be no luminescence. So at that point, when we introduce our protein of interest, if the protein is getting released, it means that they are behaving indeed like a release factor. So with this concept, we purified the protein. Under in vitro condition depleter release factor, we introduced our protein of interest and showed that, okay, when we add this protein, it has the ability to release the protein, which then we can consider them as release factors. So then what we have done is we checked under in vivo essential. What is the essential, whether it has an essential role in the Mycobacterium. So for this, what we have done is we constructed the knockdown strain via CRISPR-Cas system, which is an in-house system which we have developed in our own lab, and characterized them at phenotypic level and functional level. So to use a Cas9 system, there is a drawback associated with it, which is the polar effect. We have to check whether the 2 genes are in operon or not. So what is the polar effect? If 2 genes are in operon, if I knock down the upstream gene, there will be simultaneous knockdown of downstream gene. So in order to check whether the genes are in operon or not, we checked at a transcript level And we construct a knockdown strain and showed that, okay, in the case of both release factor, there's a downstream gene which is in operon. So how to overcome this problem? So we complemented back the downstream gene to construct release factor knockdown strains. So we validated it and we showed that, okay, when we knock down this release factor, there's a significant growth defect at OD level, CFU level and spotting. So which comes the phenotypic. So since we are claiming that this protein is associated with the translation or protein synthesis, we thought of utilizing a puromycin uptake assay. Puromycin, so in the case of actively translating ribosome, more amount of puromycin will be incorporated, and when the cell is not actively translating, less amount of puromycin will be incorporated. So with this concept, what we have done is we depleted the release factor initially, then treated with puromycin and checked what happens. So we found that when under a non-depleted condition, there's a significant amount of puromycin uptake. Under a depleted condition, there's a less amount of puromycin uptake. And we complemented it back. And showed that there is a significant uptake, meaning that the protein is associated with the translation. So next, what we have done is we were planning to capitalize the fast-growing organism, which is Mycobacterium smegmatis, as a surrogate model. So what we have done initially is the sequence alignment and checked how much is the identity, and we found that it is more than 80% identity. So, and motives are more or less conserved. So with this, and we checked at the loci level and also showed that this is conserved. Next, what we have done is we depleted the PRF release factor in the smegmatis and tried to complement it from the proteins from the RV, Mycobacterium tuberculosis, and we showed that, okay, there's a conservation function, and we constructed the GGQ mutant and showed that when we mutate and try to complement it back, there is no complementation, indicating that this motif is indeed essential for its translation. Next, what we have done is we wanted to probe the specificity of these proteins. For this, we constructed the nanolook with all the 3 stop codon variants and did prior to that, what I have done is we profiled the stop codon and showed that UGA is the most prominent stop codon. And then we validated it at in vitro level and showed that PRF-A has the ability to recognize UAA, UAG, and PRF-B has the ability to recognize UGA, indicating that PRF-B has more dominant role in MTB because UGA is the most dominant stop codon. This was done in vitro level, and then we validated the same in vivo level, where we constructed where we deplete the release factor proteins and try to express the nanolook with all the 3 stop codons and showed that, okay, the result is more or less same. So what are my key findings? There is a release factor followed by downstream genes are in operon. We were able to successfully generate knockdown strain particular to release factors. Depletion of these release factors lead to significant growth effect, and there is a functional conservation between the smegmatis and the M tuberculosis. The motif GGQ is found to be very essential for the survival. And through the in-house developed nanolook assay, we were able to validate the stop codon specificity. And now we are interested in understanding what happens when we deplete the release factors and what all are the rescue mechanisms which is associated with its survival. And we are interested to characterize the specificity of these proteins by mutating the stop codons and its in vivo survival. I would like to thank my supervisor, Dr. Nishit Agarwal, and friends, all of them, for my support in my work.
The floor is open for any question and answers.
Very nice, and I followed it really nicely. I wonder whether, taking it one step further, where you think possibly you can use this technology for treatment of TB. Is it to target the translation? What is the translation actually doing into the human cells that is causing the disease?
Sir, for example, if we are able to make a drug particular to this protein, the point is, your concern is about drug resistance, right? So let's assume a scenario where this protein is getting mutated. So in that scenario, what is happening is that fidelity of this system is compromised. So which again ends up in extending the protein. C-terminal extension will be there, which will again get degraded. Basically, the bacteria's fitness is totally compromised. So the chance of generating a resistance is also damage to the bacterium in this scenario. That is the translational aspect. So we are focused on that part also. Thank you.
Hi, can you hear me? Yes.
Yes.
I wonder if you saw a reduction in bacterial viability when you knocked out PRFA.
Sir, for example, this is an essential gene.
Yeah.
So we can't knock it out. So we can only silence or suppress the expression more than 90%. If we try to knock it out,
Yeah.
as I said, it will die.
Okay. Yeah.
So. Thank you.
Next, I invite Ms. Kajal Kamboj to the stage.
Good morning, one and all present here. I am Kajal Kamboj, fourth-year PhD student working under the guidance of Professor Bhabatosh Das. I am here to present my research work. The title is Discovery and Development of Therapeutic Bacteriophage Cocktail for Multi-Drug-Resistant Gram-Negative Escape Pathogens. So before directly moving on to the work, I would like to give a brief introduction why we started working on AMR. So antimicrobial resistance occur when a microorganism becomes resistant to an antimicrobial agent through the acquisition or evolution of resistant mechanism, leading to treatment failure and increased mortality and mobility. WHO has recognized AMR as one of the top 10 public global health threats, and this is because of the major death taking place by AMR. 1.14 million deaths has already been taken place in 2021, while the death associated were 4.71 billion. And it has been estimated by WHO by 2050, 39 million deaths will be there. So this main reason is that the bacteria are developing resistant and we are not having enough antibiotic in the clinical pipeline. In one of the data, it suggests that 2023, we were having 97 antibiotics in the clinical pipeline, which is now reduced to 90 only in 2025. So as I said that the bacteria developing resistant, you can see it from the bacterial priority pathogen list, which was released by WHO in 2017, that talks about the different gram-negative pathogens, but when the same list was released in 2024, the sequence of these bacteria has changed, but still we are having the gram-negative bacteria in the top list. So one of the reasons of selecting the gram-negative bacteria is this one, and as I said that the bacteria are developing resistant, not having antibiotics enough to tackle those pathogens, the people are moving to the other clinical approaches. And here I want to show the different clinical pipelines which are there in the development. So apart from the small molecule that constitute 46%, the bacteriophages, it account the 13% in the pipeline. The other important point is that apart from the critical priority pathogen, it also targets the high priority pathogen. So one of the reasons of selecting bacteriophages is this one. So as there are multiple needs because of increasing MDR, XDR pathogen, we are not having enough antibiotics. There are also certain infections caused by the biofilm forming bacteria. We need to move to some other approaches, and bacteriophages are the promising approach for this. They are highly specific, they are effective against MDR and XDR pathogen. They have the ability to self-amplify at the site of infection, and that's why we started working on bacteriophages. So this is the complete workflow from where we have started and how the cocktail development takes place. So we started the sample collection from the sewage, and after the collection of the sample from diverse regions, we processed the sample and isolated the bacteriophages against different bacteria. So we have used clinical MDR and XDR pathogens, and the target pathogens were E. coli, Klebsiella pneumoniae, Acinetobacter baumannii, and Pseudomonas aeruginosa. After that, the plaque overlay method was performed, and then identification and characterization of phages were done using whole genome sequencing and electron microscopy. The next step is screening of the phages, the isolated bacteriophages, whether they are narrow spectrum or broad spectrum. We did the screening of bacteriophages. And finally, from screening, we selected the bacteriophages that are suitable for cocktail development. So talking about the bacterial isolates that were included, so we first looked to the Indian isolates, which are the prevalent STs across the different gram-negative pathogen. So talking about A. bomani, the total isolates were 351 and the total STs were 16. So out of the total 16, we found that the ST2 is the most prevalent, followed by ST25, 1 and so on. Similarly, it was done for the other 3 bacterial isolates as well. We have included the antimicrobial resistant profile of the pathogens. All the pathogens that were included in the study, they were MDR and XDR, and also they belong to the different clinical sources. The total clinical isolates that were included in the study were 101. Now talking about the bacteriophages, so as I said that we have included the different regions of India for the bacteriophage isolation. So total state covered were 6, from where we have included the 15 sampling site and the bacteriophage isolation was done for the 4 target pathogens. We have included the wastewater from the hospital and also from community and sewage treatment plant to have a diverse source of bacteriophage in our collection. So these are the certain photographs from where we have collected the samples. Further, after the collection of the samples, it was processed in the lab and agar overlay method was performed to isolate the bacteriophages. You can see that there are certain zones which are present on the plate which depict the presence of bacteriophage in the sample. The next step is identification and characterization of these bacteriophages. So we went with the electron microscopy and also the whole genome sequencing to look at which particular coding sequences are present in the genome of phages. We found that there are different genes involved in different functions, and also the whole genome data, it tells that whether there is any AMR gene, virulence factor, or any other specific gene present in the genome. We also compared the whole genome similarity of the phages isolated by us to already submitted phages in the NCPI database, and we found that there are certain regions or the genes which are unique in our phages. So after isolation identification, the next step is screening of these bacteriophages against different clinical strains. So while I was performing this screening, I found that there are different morphologies of zones created by the bacteriophages. So based on the different morphology, we have characterized these bacteriophages. Like if it is a clear zone, we assign it number 1. Similarly, based on the different morphology, different number has been assigned. So here I am showing the result for my primary screening, where initially in the primary screening we have included the 8 clinical strains against which the bacteriophage isolation or the screening was performed. So as I had targeting the 4 pathogens, so against all the 4 pathogens the primary screening was performed. Based on the primary screening results, we went with our secondary screening, where we have increased the number of clinical strain, and finally we went for the cocktail development. So this is how we narrow down from the complete bacteriophage isolation to having the combination of bacteriophages. So we have collected the samples, further doing the primary screening, and there are certain criteria for a bacteriophage to be included in a cocktail, that the phages should be lytic in nature, they do not have any AMR gene or virulence factor, and also they should be broad spectrum. So having all those criteria, we finally went with the secondary screening and having the bacteriophages in the cocktail. So this I am showing for Acinetobacter baumannii. Similarly, it was done for Pseudomonas, Klebsiella, and E. coli. And finally, the cocktail activities were it was tested in the different strains. So here you can see that in the case of E. coli, the total strains I have used 21, in which it was active in 19 strain, which gave the phage susceptibility to 90%. Similarly, for all the 3 pathogens, it was performed. And when we talk about bacteriophages, one of the things is resistant development, how fast the resistance is developing. So we have also considered the resistant development in the consideration, and we found that the resistant frequency is quite less when talking about the Klebsiella, E. coli, and in the case of Pseudomonas, the resistance was coming after 48 hours. So this is, we are having the result till in vitro. Moving on to the in vivo, we found that for the bacteriophage therapy, it is the topical application which is the best approaches. And in the topical application, we went with the diabetic wound ulcer model, infection model. So we have started with the E. coli infection where we have created the wound, and further the treatment was given in 2 doses, first as the 2-hour and second as 24-hour. And we have included infection and the treatment, also the control group, where in each group we are having 6 mice. And after doing this infection, we found that with the treatment of bacteriophage cocktail, there is a significant bacterial reduction. So we are having 4 bacteriophages cocktail targeting E. coli, Pseudomonas, Klebsiella pneumoniae, and Acinetobacter baumannii. All the phages that were included in the cocktail, they do not have any AMR gene, no virulence factor, they are all lytic in nature and also target broad spectrum range. We have just did our in vivo experiment for E. coli. We have to do for Klebsiella, Acinetobacter baumannii, and also for Pseudomonas aeruginosa. We also need to check the safety and toxicity of bacteriophage cocktail. I would like to thank my guide, Professor Babatosh, for all the guidance, and my RAC member, Shrikant sir, Shushmita ma'am, for all the suggestions during my RAC meetings, my lab members and friends, my former funding from UGC, current funding from ANRF, and most important thanks to St. Newton Bandis Probiotical Private Limited. I also want to thank Vishal Sarani Manchi ma'am from FICCI. Thank you very much.
Any questions from the audience?
So during the lytic cycle of this bacteriophage replication, so there will be release of like bacterial components,
Yes.
which will work as a PAMPs. So that could be detrimental for the host. So how to tackle that?
So when bacteriophages, it lies a bacteria, there are certain release of endolysins. So we have also considered that the endolysin concentration will also be in the range as approved by FDA. And when we talk about the topical application, that particular range is quite less. But when we move to the systemic circulation, then we have to consider that it should not increase beyond the range. So we have also considered that point. So.
Okay, thank you.
Okay, hi.
Thank you very much.
Excuse me. One small doubt. So did you check, since this is a broad spectrum phage, so did you check against commensal bacteria?
Yes, so we have also looked in that particular, that whether it is having any effect on the commensals already present on the gut microbiome. So it is very specific, like it will only have the effect on the pathogenic or based on the receptors, like it's very specific. So it do not have any effect on.
Okay. And for E. coli and Klebsiella, we have capsules, which is more prevalent.
Yes.
So it is effective against encapsulated bacteria as well?
Yes, we have also that's why we have considered that ST type. So we are having the different ST types while we were doing the screening. So it was effective in most of the STs. Okay.
Just one question from me. Two questions actually. So when you said it reduced the number of bacteria, by what percentage? Was it a total kill or
So
how
it
did
was.
you?
So it was based on, sir, we have looked for the CFU burden. So it was like it is the infection on the skin. So we have calculated that how many bacterial burden is reducing on the skin.
Yeah,
on the
so
part of
by how much did the CFU reduce?
So it was, when infection was given, it was 10 raised to power 8. When we given the bacteriophage cocktail, so it was 10 raised to power 7 and 6. It was in the range of that. So there
But
is a
it was not
significant
eliminated.
difference.
But it was not eliminated.
No,
Did you
because
use a
it is-
control, antibiotic
Yes,
control?
yes,
And
it
what
was
was
also-
the reduction in the antibiotic control?
So in the antibiotic also, like you can see that here is the result, like it was in the 10 raised to power 6.
So similar to antibiotics.
Yes, almost similar.
So,
But
so the second question is, where do you see this going? How do you take it forward?
like the strains, most of the strains in the clinical study which we have included, they are antibiotic resistant. So we have included the strain for the animal experiment that are like cholesterol susceptible. So most of the strains are not having any effect, like antibiotics are not having any effect.
No, no, that's
So
okay.
there
I'm
we
saying,
can-
how do you take the bacteriophages forward so that
So-
they come into clinical practice?
Yes, like in most of the other countries, it's already been in the clinical trials, but in India, it is like there are certain companies which are working on bacteriophages, so we can use them initially for the topical application, and then once we are having like how it is behaving in the topical, then we can move forward for the systemic also, like how it is.
Thanks.
you measured the resistance in vitro, I believe, right? So when you infected mice with the, and then treated with the phages, so you said like 10 to 100-fold reduction in the CFUs were obtained. And I think this is only based on one time point of analysis. Did you follow for the successive time points and see that there's a constant reduction in the CFU? Number one. And number two, what was the resistance frequency during mouse infection? Was it similar to the in vitro or better or less?
Okay, so the in vivo experiment we have just started. So in the case of this, like you are talking about the time point, so we have only did this experiment for 48 hours right now, but we will also consider that you are suggesting. And also for the resistant, we have only did in vitro right now. In vivo, we have to do.
So I think before moving forward, 2-3 things are necessary to be done. One is these, and then thirdly is to know the mechanism of resistance and the susceptibility, I think. Yeah.
Okay, sir. Thank you so much.
Thank you, Kajal. Now I invite Ms. Jyotsna Dandotia.
Good morning, everyone. Myself, Jyotsna Dandotia. I'm from Immunobiology Lab, working under the guidance of Dr. THSTI. So in this study, we investigate how dietary ions like potassium regulate TH9 cell biology and how we can use this to improve anti-tumor immunity. So let me just brief about the adoptive T cell therapy. So adoptive T cell therapy is highly promising personalized cancer immunotherapy. In this therapy, patient own T cell isolate, expand or genetically engineer ex vivo, then again reinfuse into the patient to eliminate, kill tumor cells. So they are right now major two approaches. One is CAR T cell therapy, which has shows remarkable success in hematological cancer, and the other is TIL therapy or TCR therapy, which also showing encouraging result in solid tumor. But despite all these advances, these adoptive cell therapy showing very limited efficacy in solid tumor because of these major challenges like poor persistence of the transferred T cell exhaustion in immunosuppressive tumor microenvironment. So the challenge is how to enhance this adoptive T cell therapy. So the promising strategy is to identify the right T subset that promote potent anti-tumor immunity and to enhance this subset for better persistence within the tumor microenvironment. So our lab is working on these TH9 cell. This is a distinct subset of CD4 T cell. It's generated in the TGF beta and IL4 cytokine and characterized by the production of IL9. So these TH9 cell possess potent anti-tumor immunity by producing this IL9 and IL21, which actually recruit or activate NK and CD8 T cells in the tumor microenvironment, which basically convert cold tumor into hot tumor. Also, these TH9 cell can kill directly tumor cell through this GRAN enzyme. So all these properties making them highly promising for the cancer immunotherapy. And also there are recent study which highlight that these TH9 CAR T cells shows better persistent and resistant in the tumor microenvironment, which further highlight their therapeutic potential. So it's important to understand these TH9 cell in cancer immunity. And we have also, our lab is also working on this dietary ion, how these dietary ions regulate T cell biology and how we can use this to improve anti-tumor immunity. Our lab has previously shown that high salt diet increase anti-tumor response and reduce the tumor burden in mouse model. Also zinc can modulate this immune regulation. And there's a science paper which has shown that potassium increase stemness and persistence in CD8 T cells and reduce tumor burden. So all these findings lead us to explore can these dietary ions enhance TH9 differentiation and improve cancer immunotherapy. So we use this IL9 GFP reporter mice. We harvest spleen and lymph node. We make single cell suspension, shorted these knife T cell with the help of the shorter, and we differentiate the knife cell into the TH9 cell with the help of the IL4 and TGF beta cytokine, and we characterize IL9 with the flow cytokine cytometry, RT-PCR and ELISA. So first, the experiment what I have done, here I have checked all physiological relevant iron on TH9 cell, and we have found out that potassium is significantly increasing the iron production, and also this potassium increases iron in a TH9 cell in a dose dependent manner that we further confirm through RT-PCR and iron ELISA. So all these findings lead us to said that potassium is positively regulate iron production in TH9 cell. So next is how does potassium regulate TH9 cell mechanistically? So it's understand the mechanism so that we can find out the druggable targets. So we, as you can see, these T cell express lot of ion channel, including this Kv1.3 ion channel, which is a potassium ion channel. So these ion channel is important for T cell differentiation, activation and metabolism. So to further understand, we perform this bulk RNA sequencing, and in our bulk RNA sequencing we have found out that potassium treated group somewhere increasing this key CNA3 which encode this Kv1.3 ion channel. So we to understand this further, we use this TAE, which is a potassium ion channel blocker. So when we block these ion channel, despite in the presence of potassium, IL-9 production is not increasing. So somewhere this suggests that this IL-9 induction require intercellular potassium entry through potassium ion channel. Then we further look into the molecular mechanism, and the heat map shows that potassium is somewhere increasing the upregulating the TH9 cell associated cytokines like IL-9, IL-21, which further confirm our findings. And these IL-9, IL-21 is also important for anti-tumor function of these TH9 cell. Then we have further look into the transcription factor. Interestingly, we have found out the classical transcription factor of TH9 cell, SPI and IRF4 is not upregulated. Meanwhile, we have found out that the FOXO1 transcription factor is upregulated. So we hypothesize maybe this potassium is working through FOXO1, and also our previously labs have shown that FOXO1 is essential for IL-9 induction. So we generated this FOXO conditional knockout mice by crossing with CD4 Cre and IL-9 GFP mice, and we isolate these naive T cell and differentiate them into TH9 cell, and we have found out that potassium were unable to increase the IL-9 expression in this FOXO deficient TH9 cell, which somewhere confirm that FOXO is a key mediator of potassium enhanced TH9 differentiation. We have also checked the phosphorylation of FOXO in the presence of potassium. So FOXO is somewhere decreasing the phosphorylation of FOXO, which makes FOXO more active and produce more IL-9. Which confirm our findings. And next, how does this potassium modulate key metabolic pathway in TH9 cell? So we perform the untargeted metabolomics, and as you can see in a PCA plot, there's a clear separation between these two groups, which suggests the significant metabolic reprogramming. Then we have taken here top 25 metabolites, upregulated and downregulated, and we have found out that in a potassium treated group, these two important metabolite is downregulated. So these two metabolites belong to this polyamine pathway, and this polyamine pathway is very well studied in other T helper cell like TH1, TH2, TH17 and Treg, but not in TH9. So further understanding this polyamine pathway role in TH9 cell, we first used this spermidine metabolite, which was decreased in our potassium treated group. So when we treated TH9 cell with this spermidine metabolite, we have found out that it's significantly increasing the IL-9 production. The next we have used this DFMO, which is a highly specific inhibitor of this polyamine pathway, and it suppresses this ODC gene, which further suppress this polyamine pathway. So by using this polyamine DFMO, IL-9 is again increasing, so which further confirm our previous finding. And also it's said that somewhere this polyamine pathway is negatively regulating this TH9 differentiation. We will further confirm our finding with this targeted metabolomics, and we will also check this DFMO in vivo validation as a therapeutic target to enhance TH9 mediated anti-tumor immunity. We will also use this ODC1 knockout mice to check this polyamine suppression can increase IL-9 also. So these findings identify this suppression of this polyamine pathway, metabolic pathway, as a novel mechanism by which this extracellular potassium enhances IL-9 production. Next, we have also treated spermidine in the presence of the potassium. There also spermidine is decreasing the potassium effect. So in short, this potassium is depleting this polyamine pathway, increasing IL-9, but when we treat spermidine with exogenous potassium, it suppresses IL-9. This data suggests that suppression of this polyamide metabolism is required for potassium-mediated TH9 differentiation. So next we ask the very important translation question, that does this potassium improve the persistent and anti-tumor function of TH9 cell in vivo and enhance CAR T cell efficacy? So we generated this triple transgenic mice. In this mice, all cells are RFP positive. So we take these all RFP positive cells and we differentiate them into TH9 cell. Then we adoptively we transfer into this B16 ova bearing tumor mice, and we monitor tumor growth, and we sack these mice. Then we isolate spleen, lymph node and TILs from the tumor, and we track these RFP positive cells. As you can see, the frequency of these RFP positive cells is more in the potassium treated group, which further confirm that somewhere this potassium is enhancing the persistence of this antigenic specific TH9 cell in vivo, and this further correlate with the tumor burden. As you can see in the potassium treated group, the tumor size is much smaller than the TH9 cell, and this also the same result visible in the tumor volume and tumor weight. And we have also done the immunophenotyping of TILs, and there we have found out that in the TILs, the infiltration of NK and CD8 cells is increasing. So somewhere this potassium is enhancing TH9 mediated anti-tumor immunity by increasing the NK and CD8 cell infiltration into the tumor microenvironment. Also, we have analyzed our bulk RNA-seq data. There we have found out that the persistent stemness gene is increases in potassium treated group. Also the anti-tumor genes is also increases. Meanwhile, the inhibitory molecules is decreases, which suggests that these T cells are less exhausted. And also this potassium is somewhere reprogramming these TH9 cells towards a stem-like and anti-tumor phenotype, provides superior tumor control in vivo. And next we have also tested this potassium role in CAR T cell function. So we generated this CD19 TH9 CAR T cell treated with or without potassium, and then co-cultured with CD19B positive cell. In our CAR T cell killing assay, showing that potassium is also increasing the cytotoxicity of CD19 cells against TH9 cell against these CD19 cells. And in short, in summary, these potassium ion channel through is increasing the FOXO1 activity, which increasing the stemness gene, meanwhile depleting the polyamide pathway. So how these two things mechanisms correlated with each other, we are still finding. Also it increasing the TH9 CAR T cell function, enhance anti-tumor immunity by the infiltration of these CD8 and NK cells. For the translational work, we are thinking we will use this potassium in our T cell conditioning media to treat this TH9 cell in ex vivo. We will also generating the next generation CAR T cell by overexpress this KCNA3 ion channel. So thank you everyone. I would like to acknowledge my mentor Dr. Amit Awasthi for his support and guidance, my REC member, FACS facility, our collaborator, also my all immunobiology lab member for supporting this work, THSTI core academic section, administration and small animal facility for all the support. So thank you.
We can take some questions.
Can I ask whether this involves potassium channels on the T cells very specifically? Is there something special to TH9 T cells only, or all other T cells may have this potassium enhancement?
So all T cell have this because we have checked potassium role in TH1, TH2, even in TH17. There also this potassium is increasing IL-9. Meanwhile, suppressing their cytokines, like it's suppressing IFN gamma, IL-17. So yeah, it's very interesting.
And it's the environment of where the T cells are, where there's a lot of potassium, or is it just that the ion channel is particularly perturbed to bring more potassium into the cell?
So we treated this TH9 cell when with extracellular potassium. These ion channel take this extra cellular potassium inside into the T cell. So that's why we block these ion channel, then the IL-9 expression is not increasing. So that's why we confirm that they are extracellular potassium on ion channel. They are both related to each other. Yeah, thank you.
So I was wondering, what is the kind of potassium levels that you could see before the development of the tumor in the mice, and whether that would keep depleting, and so you need to have extraneous potassium addition so as to basically reduce the tumor? Have you looked at whether tumor growth progressively leads to reduction in the potassium levels in the animal?
Sir, so here we are not using potassium orally. We are just treated these potassium with potassium TH9 cell. Then we transfer these TH9 cell into the in vivo models. So
tumor progression is going to lead to potassium levels altering, and that's the reason you need to have potassium more so as to increase the pumping into the T cells.
yeah, yes, sir. Yes, sir.
So you probably want to look into that.
Okay, sir. We have not checked that actually, sir. So
That's okay. Thank you.
Okay, thank you, everyone.
Thank you so much, Jyotsna. Next, I invite Ms. Sneh Bajpai to the stage.
First slide. No, no. The first slide. Good morning, everyone. My name is Sneh, and I'm from Dr. Krishna Mohan Atmakuri's lab. And briefly, my PhD work is on preclinical evaluation of a maternal vaccine candidate against Klebsiella pneumoniae. Briefly, sepsis is a dysregulated host immune response to an infection, and when such an infection presents itself in a neonate, it is classified as neonatal sepsis. Neonatal sepsis globally impacts more than 3 million neonates annually, and with that, it's a major public healthcare concern, especially in LMICs, where majority of these infections are caused by gram-negative pathogens such as Klebsiella pneumoniae and Acinetobacter baumannii. Briefly, the transmission is in early onset sepsis is through the vertical transfer of infection from the mother to the neonate, mostly by ascending infection and at the time of delivery. And the late onset sepsis is mostly the community acquired infection. There are multiple strategies to reduce the burden of neonatal sepsis, out of which the most interesting and the most attractive strategy is maternal immunization, given that it not only induces a cell-mediated immune response, which prevents the pathogenic colonization in the genitourinary tract of the mother, but it also induces a humoral immune response, which protects the neonate in the early days of life. GBS was a major problem in the West, and they already have a maternal vaccine candidate in the phase 2 clinical trials, but unfortunately in India we don't have any maternal vaccine candidates against gram-negative pathogens. So in literature, multiple studies have been already published where people have used different vaccine platforms to come up with vaccine candidates against Klebsiella, but none of them have made it to the phase 1 clinical trials as well. That creates an unmet need for vaccine candidates against Klebsiella. So we wanted to look at the use of outer membrane complexes from Klebsiella pneumoniae as a vaccine candidate. Now, how does this help us? It helps us to actually direct the immune response towards the pathogen's membrane proteins rather than the cytoplasmic proteins, which are also being expressed in terms, which are also shown to the host when we use the whole bacteria as an immunogenic antigen. So based on that, we have we hypothesize that immunization of the female murine model with Klebsiella-derived membrane components will not only extend protection in to the fetus in utero, but also to the neonate against sepsis and Klebsiella-mediated sepsis in particular. So we wanted to ask three major questions: that whether the membrane components are superior or inferior to the whole cell or the lysate in terms of generating protective immunity. Following that, we wanted to know whether immunizing the dams will confer this protection to the neonates. And finally, we wanted to understand whether Klebsiella-derived membrane will it confer cross-protection against other escape pathogens. And to begin with, we wanted to first establish a female murine model with Klebsiella pneumoniae. So basically, this is a bacteremia model where we take 11 to 13-week-old C57BL6 mice, and we inoculate the pathogen through intraperitoneal route, and then we basically look at the physiological changes as the infection progresses, and we also look at the serum cytokine, chemokines, and sepsis markers, which are also looked at in clinics, in case of human infection, sorry. So first we had to identify a lethal strain. We have a collection of clinical isolates through which we had identified that KP-TRU888 is one of the most lethal isolates that we have. And using that, we also standardized the lethal dose, which will be used in the subsequent objectives as a challenge dose. Next, we wanted to check whether the bacteria disseminates across the entire body of the host. And as you can see that within 1 hour of infection, the bacteria gets into all the major organs. Following that, we wanted to look at the hematological parameters, and we could see that there is lymphopenia, leukopenia, and neutropenia as the infection progresses, which is classical to severe sepsis. Now, the reason that is happening is because if you look at the tissue pathology, as the infection progresses, the neutrophil infiltration is followed by edema, which is an outcome of inflammation. You can see it here. This thing is not working. I mean, towards the end of the infection, there is severe inflammation in the tissues. As a result of which, the organ function also reduces as the infection progresses. Following that, we also wanted to screen for the pro-inflammatory and anti-inflammatory cytokines in the circulatory serum, and you can see that as the infection progresses, the levels of these cytokines and chemokines are elevated significantly. Lastly, we wanted to look at the organ, the damage-associated molecular patterns such as calprotectin in the host circulatory serum, and as the infection progresses, again, the levels of calprotectin increase significantly. With that, we show that our model basically recapitulates all the hallmarks of severe sepsis that are seen in human patients. Moving on to the next objective, which was to enrich the Klebsiella pneumoniae membrane components. Now, for doing so, what we did was that we took the bacterial lysate and we washed it with different sequential buffer washes, and we then did proteomic analysis of the enriched membrane. Now, as you can see here, that as compared to the whole cell lysate, the final enriched membrane has a very few number of proteins, showing that the enrichment was done successfully. Then to ensure that the protocol is reproducible, we repeated it quite a number of times, and as you can see it from the banding patterns, they are quite similar across the different preparations. Then we evaluated the proteome of the enriched fraction, and we identified 207 proteins, out of which 53% was coming from the membrane proteins. And to benchmark our protocol, we compared it against what was already published in the literature, and we saw that our enrichment outdid what was already published in the literature. So looking at the proteomic data, we found out that most of these proteins were essential for the pathogen survival in the host, and we then identified some candidates from the outer membrane, from the inner membrane, and from the periplasmic region, and so as to ensure that there is batch-to-batch consistency in terms of abundance, and also we aim to use these markers for evaluating our batch consistency in the next round of enrichments. The third objective is where we actually are going to compare the protective efficacy of the Klebsiella pneumoniae derived membrane against the heat-killed and the cellular lysate. So for doing so, we have three antigens here. First is the heat-killed Klebsiella, second is the cellular lysate, and the third is the enriched Klebsiella membrane. And we have paired it with two distinct adjuvants. First is aluminum hydroxide, and second is emulsipan to see its combined effect. So to evaluate which of these combinations is actually superior, we have data from the challenge study. Following that, we'll be looking at the antibody titers, and then we'll be looking at the serum bactericidal assay for assessing the quality of the antibodies, and finally, we'll be also looking at the T-cell responses. Briefly, this is the immunization schedule that we have followed, which involves one prime dose following two homologous boosters 3 weeks apart. At the end of the immunization, we divide our mice into 3 cohorts. One remains unchallenged, and 2 of them are taken for lethal and 10x lethal challenge. So before taking these formulations into the animals, we did LPS quantification so as to ensure that the antigen itself is not toxic to the animal. And we found that the LPS quantities were well within the permissible range. From the challenge studies, you can see that heat kill plus alum in both 10x and lethal challenge have a better protective efficacy as compared to the other combinations. And we wanted to understand why is this happening. So we looked at the total IgG titers which were raised against the immunizations, and we found that as compared to the heat kill plus alum group, heat kill plus emulsipan raised higher antibody titers, but still it was not protective enough. The same was observed during, with the lysate group as well. The antibody titers were high, but they were not protective enough. So then we wanted to understand what kind of an immune response is being raised in response to the immunizations, whether it's a Th1 or a Th2 dominant immune response. So we did the antibody subtyping, and we calculated the IgG2 is to IgG1 ratio. And interestingly, we found that heat kill plus alum group is the only one which gave a mixed immune response in contrast to the other combinations. Finally, to assess the antibody quality, we did the serum bactericidal assay, which is an in vitro assay where we incubate the heat inactivated immune sera with the baby rabbit complement factors and viable bacterial cells to see the antibody, how many viable bacterial cells is antibody able to kill. And you can see again that heat kill plus alum group does better as compared to the other groups tested. Finally, for the fourth objective, we want to see whether these immunizations actually impart an intergenerational tolerance. And I'm currently doing some standardizations for that, for this particular objective. But briefly, we plan on standardizing a murine pregnancy model. Following that, we will also be challenging the pups from the immunized dams to see if the maternal vaccine candidate is actually protective. Along with that, I also have remaining 5 formulations that I need to test in the in vivo system, and also to study the T-cell response, we'll be doing a splenocyte re-stimulation assay of the top 3 candidates. Other than that, in our lab, we also have access to a prospective mother-infant cohort where we have the matched maternal plasma, cord blood, and neonatal plasma samples from suspected patients of neonatal sepsis. So we want to evaluate whether the transplacental transfer of the naturally acquired pathogen-specific IgG is protective is correlating with the outcome or not. As well as that, we want to evaluate whether the transfer efficiency differs among different escape pathogens and define how these differences shape the passive immunity. So the reason that we want to do this is because it will help us to identify the gaps in passive immunity and also generate support for maternal vaccination as a strategy. So I would like to thank my PI, Dr. Krishna Mohan Atmakuri, my RAC members, and the animal facility and my lab members. Thank you.
We can take some questions from the audience.
Sneh, yes, sir.
Yes, sir.
So what was the purpose to compare the immune response of the protection of purified OMP versus the lysate?
Sorry, sir?
You compared OMP versus lysate protection, right? What is the purpose for this?
So in literature, many groups have used heat-killed as a vaccine. So we don't have a positive comparator in the market, right? So we wanted to compare its efficacy with both of the popular choices, that is the lysate and the heat-killed bug itself. So these two are the most commonly used positive controls, so that's why.
Compare an enriched Caucasian worse. OMP is enriched. OMP you enriched it.
Sir,
Purified and like must have used at higher concentration than the lysate.
So you are saying that the enriched membrane will be more
Yeah, it will give
concentrated in
better-
terms of the membrane proteins as compared to the lysate.
Yeah, enriched protein will give better response because it will
Exactly, exactly. But then, other than that, we are also using the heat killed as a positive comparator. So we just wanted to look at whether there will be any change in the immune response or not. I mean, we just wanted to see it for our comparison. That's it. Thank you.
Thank you, Sneh. Next, I invite Miss Juhi to the stage.
Okay. What is... very good morning to one and all present here. I am Juhi, a fourth year PhD student working under the supervision of Dr. Ramandeep Singh. So today I'm going to present a part of my PhD work entitled as Analysis and Characterization of REL-BE-TA Systems of Mycobacterium tuberculosis. So I'm starting with giving a brief introduction about TB. So as we know that TB is an infectious disease caused by the pathogen Mycobacterium tuberculosis. The disease itself has been known for centuries. It's spread via aerosol and mainly affects the lungs. Approx 1/3 of the global population is asymptomatically infected with TB, making it difficult to cure. Major challenges in curing TB involves a synergy with HIV, emergence of multi and extensively drug-resistant strains, and its ability to persist in the dormant state. When MTB enters inside the host body, it has to deal with a myriad of stress conditions like pH, redox, nutrient starvation, and antibiotic stress conditions. And in order to deal with those stress conditions, it has evolved several adaptation strategies, among which TA system is one of the strategies which it employs in order to sustain its survival within the host. Now, what are these TA systems? These are basically small genetic elements composed of a stable toxin and a labile antitoxins. So what happens under stressful environment? The antitoxin is degraded, thus freeing up the toxin, which targets some essential cellular processes like translation and inhibiting the cell growth. This growth inhibition by toxin is often reversible when new antitoxins are available and the bacteria can restore its growth. Various phylogenetic and bioinformatic analysis have shown that MTB genome harbours a large repertoire of TA system, raising some key questions like why so many are there and what are the role of these TA system in the physiology and pathogenesis of MTB. So in order to answer these questions, my PhD objective is the analysis and characterization of REL-BE-TA system and to study its role in the violence and pathogenesis of MTB. MTB harbours three REL-BE-TA system, REL-BE1, REL-BE2, and REL-BE3. Recent studies have shown that REL-E1 and REL-E2 targets the anti-Child-Altano region of the 16S ribosomal RNA, while REL-E3 targets the mRNA. So here I'm going to show the studies which have been done so far for REL-BE2 and REL-BE3 TA system. Studies have shown that the crosstalk exists between the REL-BE-TA system, wherein one REL-E toxin can be cross-neutralized by other REL-B proteins. So we wanted to investigate that apart from this, which other non-cognate antitoxins of the type 2 subfamily can cross-neutralize the activity of REL-E toxins, for which we have used a library of antitoxin constructs which were cloned in an acetamide inducible vector. We have used a co-expression system in M. smegmatis, wherein the production of REL-E and REL-B proteins were done by adding the inducers ATC and acetamide in the early low phase cultures of M. smegmatis. After that, which we have done the growth analysis. So as expected, we found that when RELI2 toxin was co-expressed with the antitoxins RELB2, there was growth restoration, but apart from it, there were two other non-cognate antitoxins, which were VABY1 and VABY2, which were also able to restore the growth effect posed by the overexpression of RELI2 toxin. The similar thing was confirmed using CFU analysis as well. So we are speculating that this non-cognate interaction will fine-tune the growth of MTB rather than leading to complete growth arrest, which will be the case if non-interacting toxins will exist. Further, to study the role of RELBE2 TA system in the virulence and pathogenesis of MTB, we have generated the RELE2 and RELBE2 knockout strains using temperature-sensitive mycobacteriophages, and the replacement of a gene of interest with the antibiotic cassette was confirmed using the southern blot. We wanted to see what would be the effect of the deletion of these genes in the in vitro growth and the biofilm formation capacity of the bacteria. So we found that RELE2 and RELBE2 both were indispensable for the in vitro growth and the biofilm formation of the bacteria. Now, because these TA systems are stress responsive modules, so we wanted to see if RELBE TA system also has role in providing stress tolerance to the bacteria, for which we have compared the bacterial survival of the knockout strain with that of the wild type strain, and we found that the RELE2 strain, the bacterial load was less in comparison to the wild type strain, indicating that RELE2 contributes to the rifampicin persistence of the bacteria. Further, we have studied whether we have done the virulence study for RELBE TA system using C57 black 6 mice as a model of infection. We have compared the bacterial load in lungs and spleen for the knockout and wild type strain, and we found that in the C57 black 6 mice, RELE2 was found to be not essential for the survival. But surprisingly, when we did the virulence study in the BALC mice, the RELE2 knockout strain was attenuated for growth, while the RELBE2 knockout strain, the bacterial load was comparable to that of the wild type strain. So the possible reason for the differential growth pattern of these knockout strain in these mice from the differential immune responses of these mice, which needs to be investigated further. Further, we did the virulence study using guinea pig model of infection, which resembles more close to the humans. And here also we got resemblance which were more similar to what we were getting in the BALC mice. The delta RELE2 strain was attenuated for growth both in lungs and spleen at both the time points. While the delta RELBE2 strain, the bacterial load was comparable to that of the wild type strain, indicating that delta RELE2 is attenuated for growth in guinea pigs and the BALC mice. From here, we made the hypothesis that whatever in vitro and in vivo phenotypes which we are getting, likely because of the antitoxin and not the toxin per se, because if it's been a toxin-mediated phenotype, then we should have got attenuation for RELBE2 knockout as well, which is not the case. So we checked the levels of antitoxin in the delta RELE2 knockout strain, and we found that it was getting 3 to 4-fold getting upregulated. So we did the virulence study using the antitoxin overexpression strain, and we found that the antitoxin overexpression strain was showing less bacterial burden in comparison to the empty control, indicating that the RELBE2 is associated with the in vivo growth defects which we were getting. Now, there are studies which suggest that copper resistance is essential for the virulence of MTB, and in the recently published research paper from our lab also, we have shown that the VABC36 toxin is required for the copper tolerance and the pathogenesis of Mycobacterium tuberculosis. So we wanted to see if RELBE TA system also has role in providing copper tolerance to the bacteria, for which we have compared the bacterial growth of delta RELE2 knockout strain with that of the wild type strain, and we found that it was susceptible to copper. We have also checked the susceptibility to other metals ion, but in presence of zinc and iron, the bacterial growth was found to be comparable to that of the wild type strain. Now, to specifically validate the susceptibility of the delta Reli2 strain towards copper. We have compared the bacterial growth in presence of the copper ion chelator, which is BCS, and in presence of the copper ion chelator, it was found that the delta Reli2 strain growth was comparable to that of the wild type strain, indicating that Reli2 deletion makes MTB more vulnerable to copper. Similar line of experiments we did for RelBE3 as well. Here also we found that the crosstalk exists between RelE3 and other VAB-B antitoxins. We generated the RelE3 and RelBE3 knockout strain and found that RelE3 deletion affects the biofilm formation and also contributes to the intracellular survival and under oxidative stress. MTB RelE3 was also found to be non-essential for survival in the C57 BRAC6 mice, and it was indispensable for survival in the BALC mice and in guinea pig model of infection. Here also the increased expression of antitoxin RelB3 was found to be associated with the in vivo growth defects which we are getting, and RelE3 also contributed to the copper tolerance. So our study found that the deletion of RelE3 inhibits the growth of MTB and affects the biofilm formation. RelBE2 and RelB3 was found to be involved in the copper tolerance and the pathogenesis of MTB. Deletion of Reli2 significantly decreased the survival rate of MTB in presence of rifampicin. Our results indicate that the non-cognate TA complexes are as efficient as the cognate TA complexes and can restore the growth effect posed by the toxin. Increased expression of antitoxins, RelB2 and RelB3 associated with the in vivo growth effects. Future directions involve the analysis of the interaction between Reli2 with cognate and non-cognate antitoxins using biophysical methods like MST, SPR, and pull-down assays. Identifying that how genetic backgrounds influences the cellular and humoral immune responses, for which we are planning to do the immune profiling and host RNA-seq studies. Also, the proteome profiling of the RelBE2 and RelB3 overexpression strain. If our hypothesis is true that it is an antitoxin-mediated phenotype, then the proteome profile of these antitoxin overexpressing strains should be similar to that of the delta Reli2 and delta Reli3 strains, and the validation of the in vitro and in vivo phenotypes using complement strain. So I would like to thank my supervisor, Dr. Ramandeep Singh, my DAC members, lab members, THSTI family, and my funding agency, CSI UGC. Thank you.
Any questions from the audience?
No.
Thank you so much, Johi. These presentations reflect the depth of inquiry and innovation nurtured at THSTI. I would like to invite Eva to the stage. I would request, could you please bear with us for 2 more minutes? Thank you.
To deliver the Foundation Day lecture, we are privileged to have with us Professor Roger Fu, Vice Dean Research at the Yong Loo School of Medicine, University of Singapore. After graduating from the National University of Singapore, Yong Loo School of Medicine, Professor Roger Fu undertook his postgraduate clinical training at King College Hospital, London, and Addenbrooke's Hospital, Cambridge. For his research training, he worked in the labs of the late Professor David D. Bono, British Heart Foundation Chair of Cardiology, Leicester, Professor Maurice Brown, past President, British Hypertension Society, and Professor of Clinical Pharmacology, Cambridge, Professor Martin Bennett, BHF Chair of Cardiovascular Sciences, Cambridge, and Professor Rick Kitsis, the Dr. Gerald and Myra Doros Chair in Cardiovascular Disease, Albert Einstein College of Medicine, NYC. His research projects were funded by both the Wellcome Trust and the British Heart Foundation through fellowship and project grants. Roger serves as the Vice Dean of NUS Medicine and is the Director of the Cardiovascular Metabolic Disease Translational Research Program. Sir, we extend our warm welcome to you and request you to kindly take the podium to deliver the Foundation Day lecture.
Huge honor to be giving this lecture. Thank you very much for the invitation. It's very rare in today's world with short attention span that one is given an entire hour to speak in the lecture, so I'm holding you captive for one whole hour. And I may well have too many slides, so we will work through them. I hope to show you what Singapore is doing for cardiometabolic disease prevention, and that's something we are working quite hard on as a team. Then I also like to show you as we transition to the new phase of working with using iPSL. So spoke to quite a number of you in the audience. We have an Asian ancestry iPSL resource, and then also to inspire some ideas about commercialization. So I couldn't help but need to put a slide on to show Singapore in the middle of our Southeast Asia, and we are obviously very close neighbors. Then also to remind you what Singapore looks like, and I'm very sure many in the audience have visited us. So what you may not know is that we are a country that is mainly living in public housing. So 80% of the country lives in our public housing, which is called the HDB. And although we call it public housing, 9 out of 10 of our residents in the public housing own their apartments. And all of this is distributed into 27 towns and estates across the island. And you probably already know that we are a fairly small country, definitely by India's standards. So the country itself, in terms of healthcare, is divided into 3 corridors. And these 3 corridors look after public sector healthcare for the country. I belong to the sector that is called the West of Singapore, so that's the National University Health System. We have a medical school, and that's the school that I belong to. And there are 2 other medical schools in the other 2 sectors of this island. So going forward, what I thought to show you is how some of the things that we do in our health cluster, we think is innovating out and spinning out both commercially as well as for practice-changing innovation. So across the whole of the west of Singapore, the NUHS system, we have 3, and the soon to come, a 4th tertiary hospital, 3 specialist centers, cancer, heart, and oral health, and the 3 health schools. So I belong to the medical school, but there's also a dental school and a public health school. And then there is a whole swath of secondary care and primary care clinics in our west of Singapore. It's very important to understand the relationship between the health schools and the hospitals, because that makes the entire healthcare system commissioned not just for clinical care, which is what most hospitals have to do, but we are also missioned on research and education because of the health schools. So that also means that as a doctor in the health system, we are also broken down into different types of clinicians, if you like to call it. So some of us are very much more clinical and somewhat education and research, whereas some of us are much more clinician scientists. So like myself, I do much more research, and some do much more education. But there is a spectrum, and everyone in this health system has to some degree involvement in clinical research or education. At the country level, we are given guidelines as to how we label an individual as a clinician scientist. So in our NUHS system, there are 52 of us that are called national clinician scientists, and the rest of us are pipeline or cluster clinician scientists. Why is this important? Because if you look across the whole of the NUHS, I showed you the different tertiary hospitals and the primary care clinic. Some of us are NUS clinicians, and some of us are NUH clinicians, and you can see the total number of the clinician ratio. In fact, the entire hospital system is really not that big, so there's only 1,300 consultants across the NUHS system. And this is the spread to which how much of our time is spent doing research. So you can see that the balance is spread right through. So how do we cultivate clinician scientists in NUHS? We have such an entity called the NUHS Clinician Scientists Academy, and then also an entity called the Clinician Scientist Development Unit. So we start looking after our medical students and bring them right across the journey of research while they are doing clinical training, residency, and so on. And when they exit to become a consultant, some of us take research to become our career and profession, and that's when the Clinician Scientist Development Unit sharpens each of these individuals towards talent awards, fellowships, PhDs, and also towards professorship. So what does the NUHS system do in terms of our research? We are very much focused along Asian health. We are calling this health more than just disease. We really want to study how people should maintain their health and not just treat diseases, and not even just preventing disease, but maintaining health. Then we also take a very strong perspective on life course. So we think about each individual going from in utero, cradle to grave, and there is a whole spectrum of research programs that cover these different dimensions. And then, of course, we also pay very close attention to technology, data science, AI, bioengineering, biotech, and so on. So in the whole ecosystem, we carry out, at any one time, 600 clinical trials. Half of them are pharmaceutical-sponsored, and half of them are investigator-initiated. And not surprisingly, a large number of them are oncological. To some degree, cardiology is trying to catch up. And there is a big ambition to grow clinical trials from among the others. So we asked ourselves the question recently, how does NUHS translate our research? How do we take the discoveries from bench to impact? And I felt like this occasion, plus the conversations we had yesterday, this is a nice slide to show. So we think that we're tackling the valley of death across 5 translational pathways, one through molecular, one through experimental medicine. These are our first in human medical clinical trials. We have a lot of going on in digital health, AI tool deployment. And this is one part that we as a NUHS cluster has really done well because we are so tightly connected with our government. So whatever we are researching, we can actually bring and speak to governments so that it actually changes policy and practice. And then there's also a big attention paid to value-based care and adoption and delivery. So what can we say we have done over the years? We have brought about quite a bit of industry collaborations and partnerships, big names that you can see. We have, over the last 5 years, filed patents. We have actually 24 spin-out companies and licenses executed. So this is really giving justice to why I would be able to stand up here to speak. So some of the examples of the spin-outs are here, and all of the names that you can see, if they're very tiny, then maybe you can't, the partnerships that we have signed with various companies. So what is the country doing in terms of putting money into big programs for research at country level? One is, of course, precision medicine. So the National Precision Medicine Program, called PRECISE, has gone through 3 phases. So in phase 1, there was 10,000 individuals who were sequenced. In phase 2, which is now completed, it's 100,000 individuals from the population. And we've just now launched our 10% SG precision phase 3. So each of our clusters will get 150,000 whole genomes for sequencing. Another really big project is the one that Ashok Venkataraman is leading on NERBA, which is RNA biology and its applications. And it's a big initiative. So here they are looking at making RNA therapeutics, making it with manufacturing grade capabilities, and welcome for you to engage with Ashok as well. Quite recently, we were also given at the country level a big National AI Medicine Platform grant for us in NUHS led by Nyam. Our AI cluster sits within our healthcare firewall, so this entire GPU stacks are sitting inside, and now the country level investment sees this as a great primer, so it's going beyond what NUHS has done with a large injection. So at the end of last year, in December, we were also told of our new RIE funding. So every 5-year cycle, the Singapore government, like the NIH, puts out its research funding cycle. And you can see that year on year, there has been an injection and enlargement of the pot of money that goes into R&D in Singapore. It covers all of R&D, so it's not just biomedical. And for one aspect, they are actually going to apportion a huge amount of money to study healthy longevity. So it's looking at brain as well as physical structures of the human body, maintaining health and not just preventing diseases. So why is this really crucial now for the government? And some of the coming slides makes that point. So it turns out that in Singapore, if you live in Singapore today, you could say that you're one of the longest humans, living humans on Earth, because in Singapore, we can live up to the age of 83.9. We also have one of the best health spans in Singapore. So we live healthily till the 73.6 years old. But that still leaves a gap of 10 years. And this 10-year gap in between of ill health and chronic health is concerning the government very seriously. So the chronic disease, the reason why people can live to this long is because we have good healthcare system in tertiary hospitals to sustain the health. Dialysis, treatment for diabetes, stroke, amputations. So all of that needs to be compressed. And ideally, the government wants to extend health span for as much more as they can. So because of that, at a population level, they really have pivoted and focus their attention into the community and into population, and even made a strategy in the announcement of the $37 billion research funding, a strategy that focused very squarely on healthy longevity and population health. So health span has not caught up with lifespan in Singapore. There's a gap of 10 years. There is an inverting of the age pyramid in Singapore. By year 2050, 1/3 of Singaporeans will be above the age of 65. And then I think you also know, this is very frequently mentioned, that in Asia we have something known as the skinny fat phenotype, where we're thin on the outside but fat on the inside. So all of that is compounded by the scary statistics that tells us that although in the West, stroke and MI incidence is declining, but in the East, in Singapore at least, we are seeing rising incidence and prevalence and numbers for myocardial infarction and strokes. And the problem is really outsized because the risk factors, we believe, are all hidden under the iceberg. So to that end, we had a funding opportunity which we call Project Reset. So in Project Reset, we are really tackling population health and looking for hidden disease within our population. So the ambition here is to carefully construct a cohort of apparently healthy Singaporeans and uncover missing biomarkers and risk factors, and actually also throw in digital biomarkers because we think that lifestyle is a huge opportunity that we can grab while we are doing such a cohort study. And at the same time, we decided to also pull in other organs to study. So here's the infographic of Reset. The ambition was to collect 3,000 individuals, no past history of stroke or heart failure, so everything under the iceberg, but to some level, moderate to high risk, so individuals have to have a risk of 5% or more over 10 years. And then what we do is compile their serial phenotype. So cardiac, everybody gets a coronary artery angiogram, calcium score, vascular scan in the carotids, the femoral. We also do a liver fibroscan. We're looking for fatty liver, brain MRI, metabolic profiles, and a Garmin smartwatch. So we collaborated with Garmin. And the objective is to look at associations, trajectories, target discovery, and then there's some experiments using cells and animals as well. So we believe that when we come to the point, we will have a huge universe of data. There is a sufficiently long follow-up of 5 years with several visits in each one of these years. So right now we are at 2.5 years into, or 2 years plus into the program, and we're going to hit our 3,000 very soon. There's a huge biorepository of samples, and I'm open to collaborate with individuals who might want to work together. We have plasma, Buffy code, PBMCs, serum, urine, and stools as well. It's interesting to go through some of the profiles of the participants that we have recruited. Half of our cohort lives in private housing, and the half that lives in public housing live in the biggest public housing. So this is really striking because it's really a high socioeconomic group that have stepped forward to be recruited. A lot of them have good income in the household, and you can tell that they are high SES group because when we invite them to come for their study visit, they're all on vacation during summer. But despite that, a lot of them have hypertension. So remember that this is a moderate to high risk group. So a lot of them do have hypertension, strong family history. You'll see the lipidemia as well. But interestingly, although they are high SES, a number of them are not treated to target. And the ones who are not known to be hypertensives by their doctors seem to have some with hypertension. So they're actually undiagnosed hypertension in this group even. A lot of lipidemia, as mentioned, and quite a number of them are not treated to target. And the ones who are not known to be lipidemic actually are lipidemic. Remember, this is a high socioeconomic group, and they've not brought themselves forward for a regular screen from what looks like. For people with acceptable cholesterol, there are actually individuals who have quite a wide range of other risk factors, inflammation, fatty liver, LPA, and so on. And metabolic dysfunction is the one that we are most worried about, and it's most alarming from what it looks from our data. So we decided that we would limit the number of diabetics that we will recruit, because if we say that we open the doors for diabetes, it's very easy to fulfill the Q-risk score. So we say we will stop at only 300, which is 300 out of the 3,000. So even then, the diabetics are not really treated to target. Then among the ones that are not known to be diabetic, we find that there is diabetes hidden within them. But I think the most seriously worrying is the pre-diabetes, and the amount of pre-diabetes is very, very striking. So we're not talking about individuals who have diabetes. People with pre-diabetes are people with high fasting glucose or high fasting or high HbA1c. When we've looked at individuals with normal fasting glucose or normal fasting or normal HbA1c, there are individuals who have very high fasting insulin, and individuals with normal fasting insulin and normal fasting glucose can even have a huge insulin response to an oral glucose challenge. Although the glucose response is normal, the insulin response is sky high. So if you add up all the numbers, there is quite a lot of hidden pre-diabetes or pre-pre-diabetes under the iceberg. So the picture of Jia Yu is missing in this. It's covered by me. So Jia Yu is a research fellow and cardiologist from China. He's taken 100 of our cohort so far and done a latent class feature unbiased analysis. And he's found 5 groups of individuals. So in the first group, these are individuals with good metabolic profiles. They are not obese. This is the BMI. They have hardly any liver fat. Truncal waist circumference is also good. We did a DEXA scan, so we could also measure visceral adiposity. And their insulin and glucose all looks good, and so is their blood pressure. And this is only 23.6%. At the far end of the class is a group which is about 18%. These are the people that you should be most worried about because they have high obesity, waist circumference, visceral adiposity through DEXA scan, also liver fat in the liver, and insulin sensitivity is not great, and so is blood pressure. The other three groups in the middle are also interesting because this group has hypertension, but their metabolic features seems good. This group 3 and 4 are interesting in themselves because they seem to have uncoupled between metabolic profile and body morphometrics. So class 3 is lean by these measures. So they are not obese, their BMI seems normal, their liver fat on this scale seems okay, and for visceral adiposity and waist circumference, they're also okay. So by measures, they are lean, but they're insulin resistant. And these individuals are by measures obese, but they seem to be still insulin sensitive. So what we think is that there is a hurtling trajectory for each one of these individuals. And I think most striking is the fact that 80% of them have some level of metabolic dysfunction. So the reason we think one of the key reasons we were able to pick up all the hidden metabolic dysfunction was because we measured fasting insulin and HbA1c, which is not often measured in all screening processes. The other interesting finding that we also saw is that LDL is completely independent of all of these metabolic profiles. You can see that in this group that is metabolic preserved, they even have high LDLs. So metabolic syndrome at the moment in clinic is described as a cluster of these 5 factors. And when someone is not with elevated glucose, fasting glucose is normal. They are literally, if they haven't got any of these others, then discharged with no more further need for follow-up. So one of the things we also discover is that if we apply those metrics to our cohort, we're actually missing out quite a number of people with metabolic dysfunction. The other is a recent obesity commission that came out of the Lancet, where they divided obesity into a preclinical phenotype and an active clinical phenotype of obesity, and using waist circumference or body fat. And we are concerned because in Asia, even waist circumference is not going to be good enough to pick up individuals with metabolic disease. So yes, there are individuals who are metabolically unhealthy with normal weight, and they're also seemingly from this description, metabolically healthy individuals who are obese. But we see that in our cluster, if we classify them as metabolically healthy and obese, they're actually nearly 50% of them are not healthy, but in that description considered healthy. And then if we looked at our class 3 or class 4, which is where we see the hidden metabolic dysfunction, there are at least 78% or 68% of them that would be missed by current metabolic syndrome criteria. And we think that the 5 classes are valid and credible because there are proteomic differences in the plasma between each one of them. And there is sufficient differences between the protein classes that we see in our plasma. What Jia Yu then did was to devise a decision tree model and projected the decision tree to a Chinese cohort and the UK Biobank. And he saw that there was differences in outcomes for MI as well as stroke in the composite hazard ratio for each one of these 4 clusters that look like they have metabolic dysfunction. So we think that using deep lenses, we are unearthing more metabolic disease that is hidden among our population, especially in Singapore. We think that using traditional lenses, there's quite a bit that is missing, and we're trying to get deeper into being able to articulate these differences even more clearly. So in the meantime, I mentioned that we've also been doing coronary artery calcium as well as coronary artery angiograms. In the group that is here, half of them have fatty liver, you can see, and 69% of them have mild to severe coronary artery calcium score. We deployed a Montreal Cognitive Cognition Assessment Score. This is a questionnaire that you give to your patients to assess for cognitive impairment. And we find that there are individuals with clinically significant cognitive impairment that needs to be referred to a clinician, and some individuals who look like they're also even on the borderline. And these are all unknown to them. So here we move and pivot to the basic science. So one of the big opportunities for a cohort like this is we are really biobanking. And what we can do in the lab today is to make iPS cells, so induced pluripotent stem cells, which I know some of you use in your labs here. So Mick, Wei Chang, and Matthias, they are PhD scientists, A-star scholars. Using our deeply phenotyped cohorts, they are able to take the PBMCs from each one of the individuals that we have recruited and reprogram them into stem cells, so we have a repository of it. We're also reaching out to various Southeast Asian partners across Malaysia, Indonesia, Vietnam, and Thailand. And then we are working on disease modeling using these iPS cells, and I'll show you some of these examples, and digital twin studies as well as precision medicine. So we take the iPS cells and we can differentiate them to cardiomyocytes. We can also do protocols for endothelial cells, fibroblasts, and macrophage. What Mick does, picture is hidden from view, is combine these now to make up little organoids. So I think some of folks in the audience are also doing this. The organoids are contractile. We can instrument into the genome. We have these things called genomic safe harbors, which we published and made public in eLife. We insert these reporters into the safe harbors, and we're able to track MYH6, calcium signaling, and in this case, ATP as well. What Ke-Ying has done is to perturb the 3-cell type and the 4-cell type organoids with metabolic stress, and we see the changes, and we're mapping these changes to understand biology deeper. What Brendan has done is an experiment that we are calling iPS Cell Village. So in iPS Cell Villages, we throw in 100 to 200 people's iPS cells onto a dish, and we direct the differentiation down towards, let's say, cardiomyocytes, and we study the cardiomyocyte cell type at scale on the dish, 200 individuals. So here's the example of what they do. Both Ke-Ying and Brendan have been using metabolic stress cocktail, and we see that the metabolic stress cocktail really does reproduce lipid deposition, changes in pyruvate and fatty acid oxidation. And here's the cocktail that we're using, and we hope to publish this data and protocol soon. So as a team, what they do, as mentioned, is that they throw in 100 to 200 patient cell lines, pulling it into a village, differentiating it into cardiomyocytes, chase them with the metabolic cocktail, and then isolate them and study gene expression profiles using single-cell sequencing. And what Brendan is doing also is validating that indeed this is an in vitro proxy for the drivers of conditions such as HFpEF, preserved ejection fraction heart failure, which is something that we believe is very metabolically driven in our context of cardiac disease today. We also see that there is some level of metabolic inflexibility when we've thrown in this metabolic cocktail, and the lab uses a lot of seahorse assays, which I think also in this institute you do. So in the meantime, we are busy deciphering out the data that we have from our single-cell sequencing. We have been using a lot of algorithm and different tools to demultiplex the individuals, and looking at QTLs, expression QTLs, overlaying these QTLs with GWAS studies. One of the big ambitions and dreams is to look for targets that could be used for treatment. So what has happened to this is a spin-out. So Junit has come with me to visit your institute. Wei Chang was mentioned in the team, and we have a successful spin-out expert, Jia Hao, who comes from Cambridge, who has successfully exited his company. So he's joined us to assist us in this so-called human-first drug discovery engine. We are taking biology based on the cells that we have, building up models, using the data we collect from the cohort, and really pushing out towards drug discovery. And we believe that one day we can work together with partners to achieve the goal for personalized medicine. So summary, there is an early metabolic risk factor alarm bell that is ringing in Singapore. We're really in need of intervention if we want to close the gap. No wonder individuals are running into disease in the last 10 years of their lives. We think that cell villages and organoids is a great way to study genetics on the dish, and we'd love to partner up across the Asia region. And we have a spin-out company, and we're wishing ourselves best of luck. In the meantime, please do come and visit us in NUS, and thank you very much for listening. I guess I'm happy to take questions too, if there are any.
Hi. Hello. So very fantastic talk, very enlightening, and your work on many patients with big cohorts is really fascinating. The question I have is, like, did you have a chance to look at the differentially expressed molecular signatures between lean and obese patients with defined cardiometabolic features?
Yeah, that's exactly what we are doing right now. There's a lot of signals. We're trying to find the best story to tell from all the different signals. So at this moment, the trouble we have is that we can't define causality. It's really only association because everything is in one cross-section of a time point. So in each one of those clusters that you saw, where the lean people are also insulin resistant, and the people who are obese who are fairly insulin sensitive, we do have their biobank plasma and tissue, and we've done molecular profiles, and we're looking through the signatures. I just don't have the exact story to tell you at this moment.
Thank you.
Thank you.
Great presentation. Hello. So my question is, like, when you're dealing with precision medicine, you have like multiple options to look at treatment approaches. So how do you, take different drugs for, analyzing the response with respect to, different patient population stratification, and looking at, do you integrate like various clinical variables into your omics data? Could you share a little bit about that?
Yeah, that's a great question. I'll try and answer that and see if Karthik agrees with me. So in our world right now, for heart failure, for example, we use ejection fraction to divide patients into whether it's reduced ejection fraction, preserve, or some people even call it mid something ejection fraction. So it's really just on the contractility, but we realize that definitely for preserved ejection fraction is a very heterogeneous whole group of patients. And there is even not just metabolic drivers, but inflammatory drivers as well. So we wonder whether the day will come when all the clinical leaders will help us to stratify heart failure into subtypes. And we can offer some of these molecular signatures for the stratification. And then the drugs that target those specific drivers can then be more tailored towards these individual classes. So that's an example of the precision that we can go. Another level of precision, if you allow me to also mention, is the country thinks about prevention by behavioral lifestyle changes too. Because we also think that it's not just about giving everyone a GLP-1 injection, and you can't afford that at a country level. So we are giving devices, wearable devices, to a lot of the population, and we're trying to pick up signals to see if there's different drivers of lifestyle are more predominant in different individuals. For some people it could be sleep, some people it could be just inactivity more, some people could be the diet choices, some people could be all. Then you have to pick which is the lifestyle to personalize. That's another personalization that we're trying to.
Thank you.
Okay, so nice talk. I think you partially answered my question. When you pick up your cohort, how much the lifestyle and what kind of food intake they have, how do you control or normalize that part when you analyze your data?
Yeah, so we can't do that very thoroughly for all of the 3,000 because even to get them to wear the Garmin watch was not easy. the adherence is only about 50%. We started working with Aura Ring, and the ring adherence is 90%, so that's also itself quite interesting. And something that I also discovered in Singapore is the population in this cohort tends to be in their 60s, because when you get older, you fulfill the criteria more easily. And they really love these devices. So we really have a very high SES group of people that want to wear everything they can and understanding everything about themselves. So we are breaking them down into the different lifestyle groups.
Yeah, I have one question. So nice talk. So that's what IPSC lines you are making, like 125 villages, like that. Just probably I missed that. Like you call it 125 villages. So do you consider, because Singapore is like multi-ethnic, like a lot of Malays, Chinese, Indians origins are there.
So 125.
125 IPSC lines you are going to mix it
I
and make it as a one cell line. Like.
Yeah.
So do you consider like different ethnic origins also, like Tamilians, Malaya and Singapore?
Yeah.
Huh?
Yes. So we like to call Singapore multi-ethnic, but really it's only 3 ethnic. And for Indians, it's mainly also South Indian, so it's not really fair. And for Chinese, it's mainly South Chinese. So I don't know how diverse and heterogeneous the 120 lines are. But in the 2000, it's also majority Chinese. So we're trying to craft another project, which we will call Reset Scale, where we hope to scale into the ethnic groups and also the lower SES as well. But yeah, the diversity is really crucial. Thank you.
So the different cell lines you mix together and say, call it as a one line, and you differentiate into cardiomyocyte, and you can use it for the disease modeling or drug discovery application.
Yes.
That's what your goal.
Yes.
Not the 125 cell lines you are differentiating into and you're testing it.
No, yes. So we take the 120 people's IPS
together.
cells and we throw them together and we differentiate them at the same time.
Yeah. Thank you. Thank you very much.
Hi, Roger. Hi, Roger. Nice talk. So I want to know the epigenetics part. So basically, these cardiometabolic disorders are considered with aging. This is a great risk factor. But when we see at the aging population, this epigenetic part comes into play. So basically, when you did this reprogramming of PBMCs into iPSCs, so probably that information might not be taken into account. So how do you plan to bridge that gap?
Yeah, agree. So there was a project that a colleague of ours in Singapore proposed, where he said we will take the iPSCs from a group of diabetics and iPSCs from another that is non-diabetic, and then study them on the dish, where the heart function and so on. And actually the diabetics, they don't recapitulate. And then the non-diabetics also, they are not faithfully recapitulating. So for me, I firmly believe that what we're studying on the dish is genetics, not the epigenetics. Because the idea of making the iPSCs, you erase as much of the epigenetic marks as possible to make the stem cell. So interestingly, some of the healthy iPSCs that we made come from individuals with high genetic predisposition to diabetes. So they led very good lifestyle, so they're not diabetic, but their cells behave like they are diabetic cells. So this is a really nice validation of what I think. Thank you.
Roger, one of the things in your slides you mentioned Singapore has a high life expectancy. So I think there is a correlation with how the precision medicine is, like how you consider also environment around you as a key factor when you give a personalized approach about, looking at treating patients, like genetics and looking at lifestyle and environment. So does that also correlate with high life expectancy when we're talking about precision medicine, as opposed to any other countries which might not have the same conditions?
Yeah, it's a really interesting question. I haven't quite squared up yet because we are not one of the countries that spends a lot of money on healthcare, but yet we manage to keep our patients and population living long, very long. So I firmly believe that the reason we are able to live very long is we have good healthcare that sustains them. as I mentioned, there's a lot of dialysis that goes on. From my own family and looking at the elderly, 2/3 of them are on dialysis, and they extend their lives for 5, 10 years just because the dialysis is available. We have dialysis centers in our HDB estates. In the block of flats at the bottom, you will find dialysis centers. So dialysis is an example, amputation is another, medicines for diabetes, management of stroke, a lot of rehabilitation. We're very good at sustaining people to live longer. But what we really need to do today as a nation is to even prevent and keep everyone healthy and stop from them from moving even into this space.
Thank you. Roger, that was excellent overview. I just wanted to ask you about the larger cohort that you talked about for 10% of the population. So what is the strategy there? I mean, is it a healthy population cohort, or is it a cohort? Are you going to follow them up? What is the strategy?
So nice to ask that question, Kartik. It's really the vision of the country that they set for the longest of time, debating how they want to do that. So 10K was very opportunistic. At the time, sequencing was brought into Singapore, the machines were there, we grabbed the opportunity. Then we saw that we should go to 100K. So the 100K was population cohorts. 80,000 of them were cohorts that were already collected over the years, so the samples were sitting in the freezer. Take them out, data is available to make associations, and then all they needed to do was recruit another 20,000, very cross-sectional. And then for the third phase, the leaders have decided that they would throw the three clusters 150,000 each. And the clusters now decide what patients they want to send for this whole genome. So the clinicians are now coming together to decide whether they want to go for cardiac diseases, metabolic, cancer, rare disease. So 150,000, some of which can be prospective, probably a lot, and then some of it also retrospective, sitting in the freezer to take the opportunity. And then the hope is to use the electronic medical record, so we're all on an EPIC system, in NUHS at least, to be able to match the sequence to EPIC, so that we can use EPIC to also follow them up if they continue coming to the public healthcare sector.
So it's not one overarching project, it's multiple projects embedded.
Multiple projects in the three different clusters. I think we are all taking slightly different strategy. And then even within our own cluster, the cardiac might be quite different from the rheumatologists and from the respirologists and so on. But as an ecosystem, we've really had to help to build that platform so that the data flow can come through, and then it's safe enough within the firewall to do the associations between genomes and clinical conditions.
And just one more question on that. So is it going to be a centralized kind of arrangement, or is it going to be decentralized?
The sequencing is done centrally. So there are collection points where samples will be picked up, and then we send their samples to them. They will do the DNA extraction and sequencing, and then they keep the raw data, but they flow data back to the clusters as well. And we have to deal with our own storage from there.
Thank you.
Hello, great talk. I wanted to know about the EQTL analysis that you mentioned, and you also mentioned that plasma proteomics was also conducted. So did EQTL analysis help in stratification of the patients? And if yes, on the same samples, was plasma proteomics also referring to the same set of proteins that you could find in the EQTL analysis?
Oh, wow. Okay, so that's a great question. I haven't made the connection between the two yet. So the EQTL is because we see great value in the fact that these cells have each one their own genetics. And we are also aware that in Asia, some of the variants that we have are very unique locations compared to Caucasians. So QTLs that have been done in the West are all centered around the genes where the variants are found. And if we drive hard at the QTL in Asia, we may surface up some new genes that could be related to disease when we overlap them with GWAS studies. I haven't yet thought about linking it to the plasma proteome, but that's a great idea.
Thank you.
Hello? Hi. Excellent talk. Nice to hear this entire compilation. What was the purpose of making this genome sequencing? Is it disease correlation to the entire population, which you have been 100 million to 450 million? How you are going to make? What was outcome of 100? Then you are going to 450. What was the outcome of that?
Three purposes that I can think of. One is that we think that there is still a need for the country, because of our own ethnicity, to have our own genetic scan and understanding if we want to build up things like polygenic risk scores, prediction for diseases, and so on. I don't know if this would ever deliver, because around the world we also haven't seen that genetics is playing such a role yet. The second is that it's economy. So once we said that we are doing 450,000, all the pharma is now lined up to speak to Singapore, because they also want to look for targets for validation of their drugs. We talked about personalizing and segmenting the country. So this opportunity became really crucial for us to go into this innovation with pharma companies and so on. There is a third reason, and I've now forgotten it. Patients. So we think that we want to put genomics into the clinic and use for patients. So this is the opportunity to do that.
Translational genomics.
Exactly. For example, newborn babies, rare diseases, cancer sequencing. So not just the polygenic risk scores, which we need to build up and prove with our own Asian genetics, but actually using it in clinic for patients that need genomic sequencing.
Basically, that information required for the clinician to a diagnosis of the patient. When comes to this, you repeat the sequence.
Yes.
They correlate with the disease.
Yes.
Treat. That was
Yes.
the purpose.
Treatment decisions. Also pharmacogenomics.
Yeah, pharmacogenomics. That's right. Thank you.
Any more questions? Dr. Fu, I had a question. So you talked about a cohort, if I'm understanding this right from birth all the way till death. So what is the size of this cohort and what is the plan for the future?
Yeah, so thanks for pointing that. I realized I didn't have that specific slide. So if that slide was shown, it would have shown you a pregnant mother, a baby, and all the way to old age. And there are at the moment in our school and health system 10 translational research programs. Cancer, cardiometabolic, we have one called longevity, digital medicine, synthetic. So each program covers different sectors. So I wouldn't say that it's definitely not one cohort. I think the PIs for all the different sub-cohorts will punch me if I say this is even a NUHS cohort. Each one of them have ownership to one space in the age range. And then we try to share data so that we can exchange and study and make things even more meaningful.
What's the size of the cohort?
So there are altogether 32 groups altogether. So for ours in Reset is 3,000. Our age range is between 40 to 70. I don't know the total number. It could be around 100,000 or so.
So I was wondering about the cohort where you're monitoring the same individuals across age.
Yes.
Is that what you're doing for the 3,000 individuals, or these are individuals of different ages that you are simply...
Yeah, they're individuals of different ages belonging to different professors running. For example, the babies are going through a different protocol.
So you don't have a cohort where you're looking at right from the birth till the death?
No.
If I said so, the same individuals?
No. If I said so, then someone will punch me. Yeah.
Thank you.
Thank you, Professor Fu, for the enlightening and thought-provoking lecture. Now I request our Executive Director, Professor Ganesan Karthikeyan, to kindly felicitate Professor Roger Fu. There are a few important announcements before we break for lunch. As we break for lunch, we invite the dignitaries and industry representatives to kindly visit the central stalls located at the foyer region outside the auditorium. We request that volunteers to assist the dignitaries to their respective dining areas. Please note that for convenience, the lunch areas have been segregated into 3 color codes. Kindly be seated. The blue-violet card holders are requested to move to the Knowledge Resource Center, 3rd floor, THSTI building. The green-yellow card holders may please move to the Discovery Terrace, 3rd floor, THSTI building. And the red-maroon card holders, please go to the auditorium terrace for lunch. Kindly reassemble in the auditorium by 2 p.m. so that we may resume the program on schedule. Thank you and enjoy your lunch.
Check, 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 check.
Check one, one. Hey. Check, check one, one. check, one, 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 hey, check, check, one, one, check, 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 one,
Recording stopped. Recording in progress.
कह रहे हैं सुनील जी? नहीं भाई। मंच पे आमंत्रित करना है दीपक जी को।
check, one, check, one, check, check, one, sound testing, check, one.
Hello, check. Check, one.
check. Check.
Kindly assemble inside the auditorium. We'll be starting shortly. Welcome back, everyone. We hope you enjoyed the lunch and made the most of today's networking opportunities. As we resume today's session, we hope the discussions over these 2 days help us build lasting collaborations and impactful partnerships in the days ahead. We now invite our industry delegates to give a short presentation about their company. We invite Dr. Mrinal from Syngene. And we want to inform that the Honorable Minister is expected to arrive. In case Honorable Minister arrives in the middle of the session will come to a stop. Yeah. Yeah. Later. So presenters, please note that each presentation will be of 7 minutes. A bell will ring immediately after 6 minutes to assist you keep the track of time. Please,
Thank you. Good afternoon, everybody. Unfortunately, I don't have a presentation at the moment, but I would like to spend a few minutes outlining what our company is all about. And you can please feel free to stop me anytime if you have any questions. It'll be a pleasure for me to take them. So Syngene basically is a CRDMO. We are one of India's largest contract research, development, and manufacturing organization. Our services span across drug discovery, drug development, drug manufacturing, and life sciences. We are headquartered in Bangalore with capabilities and facilities spread across India, as well as our own manufacturing facility based out of Baltimore in the US. We are a total of around 7,000 scientists as we speak today, and we cater, the way our services are structured is to cater across the entire life sciences domain. What I mean by that is catering to not just large pharma, innovator pharma, but also to biotechs. There are services that we offer to the agri-chemical industries, and of course to the Indian generic and the other pharmaceutical companies as well. So across drug discovery, biology, chemistry, and manufacturing, our services are structured more towards bringing in innovation for drug development. I will strongly encourage all of you to kindly take a look at our website. You will find a lot of interesting and innovative work that is happening within Syngene. We are part of the Biocon group, and with Immunil as well, we form a part of the parent company of Biocon. There's a lot of innovative work which is happening within Syngene in terms of drug discovery, not just limited to discovery, but also in terms of development. The unit that I take care of is translational sciences. It is a part of the development ecosystem within Syngene. We carry out a lot of innovative work in terms of early phase clinical trials and late phase clinical trials coupled with translational work as well. And it's a pleasure to be here. We are looking forward to this collaboration with THSTI. It's an honor for us to be associated with this organization. There's a lot of interesting work that we hope to establish in the coming years, especially in the early phase clinical trial ecosystem and in the phase 1 domain. Majorly with first in human. That's the objective that we are moving forward with. Not limited to first in human, but across the early phase as well. So this is a very short introduction of Syngene. I would love to spend more time if any of you have any questions or any further information required. It will be a pleasure for me to elaborate further. Thank you. Thank you so much.
Thank you, sir. Next, we invite Dr. Anil from Biocon.
So good afternoon. So I represent, as after Brunal, who's from our counterpart in the research division. I'm from the Biocon. As you all know, Biocon is one of the premier biotech company in India. We are one of the top 20 employers in biotechnology, and the top 5 biosimilars sold in the US are from Biocon. To start with, this company started all the way back in '78 by Kiran, and now we are standing with 16,000 plus employees and 1.9 billion revenue in a year. I just want to take you through our milestones. It started way back in '78 as an enzyme company, and then afterwards we started moving towards manufacture of API using fermentation technology. Probably we are the first to do that in India. And we have many firsts in the industry. Ours is the first in recombinant insulin manufacture in India. As of now, we are one of the biggest insulin manufacturer across the globe. I think we are serving about 1 in 5 patients are receiving insulin from our company all over the world. And we started getting into the novel molecules. In 2013, the first in class CD6, anti-CD6 molecule, itolizumab, which was launched in India, is the first for India. And for a biosimilar, trastuzumab was launched in 2014. This is also industry first. And again, in coming to biosimilar, interchangeable, Glargine is ours is the first to be launched in US. And we reached a revenue of 1 billion by 2024. And also we launched interchangeable liraglutide in UK in 2025. This is the first for any Indian company to do that. As Brunal said, we also have manufacturing site across India, and we have it in Malaysia and also in the US. We have 7 manufacturing sites all over the world with total more than 16,000 employees. As we go up the value chain, we just have to move from generic drugs to biosimilar to innovation. That's where we come to collaboration with THSTI. I need to tell all this happened in the last 3 months. One call from THSTI, visit to the THSTI with the able leadership of Dr. Karthik. All this happened in short time. We look forward to working with the THSTI in many ways, innovation, especially on biosimilars and also novel molecules, early drug development, animal testing, and also human research. Thank you.
Thank you, sir. The floor is now open for questions for representatives of Syngene and Biocon.
Good afternoon, everyone. It's a pleasure to be here again, and very grateful to THSTI, to Dr. Karthik, and looking forward to the incredible partnership that we at Immunil have forged with THSTI. To introduce ourselves, my name is Amit Mukim. I'm the CEO for Immunil Therapeutics. We are one of India's CAR-T companies with a purpose to make CAR-T affordable as well as accessible not only to India but for the rest of the world. And we want to build a company both for India and from India for the globe. My colleague, Dr. Lakshmikant. So I'll introduce Immunil for a few minutes, and then I'll hand over to Dr. Lakshmi to share about the exciting partnership that we have forged with THSTI, which will hopefully further both the science as well as the innovation in this country. So Immunil was formed 7 years ago by three very eminent individuals. Dr. Kiran Mazumdar, of course you've heard of her. Dr. Siddharth Mukherjee, who's a Pulitzer Prize winner and a professor in Columbia University. And Dr. Kush Parmar, who runs a biotech fund called 5AM Ventures. The entire premise of setting up Immunil was to bring novel therapies within the reach of patients across the world who could not afford it. As we all know, 60% of the burden of most of these debilitating diseases are in the part of the world where less than 10% of the innovation takes place. And it's very important that we all step forward to solve for this. And there are three parameters that we are working on. One is, how do we bring cutting-edge science, the best constructs, the best CAR-Ts, into the country? The second is, how do we build a process as well as technology that can scale this and make it affordable as well as accessible in terms of volumes? Because as we all know, many times technologies are available, but they are not scalable. And that leads both to restricted access as well as high costs. And the third is, how do we expand this as know-how, not only for work that we do, but help in contributing to an ecosystem, which is where our partnership with THSTI comes forward. We have one CAR-T that we have commercialized. We have treated close to 150 patients now for non-Hodgkin's lymphoma. We have partnerships now with close to 100 hospitals across the country, and as we speak, we are now bringing in more programs into India and expanding beyond India. Dr. Lakshmi will share a few details about our partnership.
Yeah, hi. Thanks, Amit. Hi, everybody. I'm Dr. Lakshmikant. I'm the Chief Scientific Officer. Over the last 1 year, we have forged some collaborations with THSTI in bringing a new technology for CAR-T where it could be considered as off-the-shelf CAR-T. Currently, most of the CAR-Ts are autologous. That means patient-derived cells have to be harvested and modified, whereas we are trying to develop a gamma-delta T-cell based allogenic CAR-T, for which we jointly applied for a PRIP grant, and we were awarded the grant. And as part of that, Professor Amit Awasthi's team is working with us for developing some of the preclinical models. And with the success of this particular program, we have now forged further collaborations to take some of our other platforms, such as rapid manufacturing CAR-T, as well as we have some few clinical trials ongoing on developing the multiple myeloma CAR-T as well as for ALL. We would like to establish a GMP facility that THST has built and qualify that jointly to make sure that clinical trials can be done from here. Along with that, we are also trying to develop some of the critical raw materials that are needed for developing CAR-T, such as lentiviral vectors. And we have a patented platform process with suspension bioreactors that we would like to bring here, so that not only will these GMP LV be used for our programs, but will also open up several other programs within THSTI and outside in the academic field, so that GMP grade global quality LV can be accessible to all the researchers who are working on cell and gene therapy. I see this very opportunistic for both of us, because as an academic center which has the infrastructure and government funding to set up some of these labs, we bring in the value proposition of running some of these GMP-based facilities using well-established protocols and SOPs that we have already validated with in our Bangalore campus. And this will enable not only setting up and maintaining the GMP facility for our purposes, but will also open up these facilities for other academicians and researchers who would like to use this. Because as we move forward, I think this capacity not only would actually expand, and more researchers can then utilize this facility. So I look forward to this collaboration, and thank you both Dr. Karthikeyan and Amit for giving this opportunity to collaborate together. Thank you.
Thank you, sir. Next, we invite Dr. Neelima Danpat from Firm Box Bio Private Limited.
Firm Box, no? Yeah.
Good afternoon, everybody. Very happy to be here. There's a little connection with Biocon. I worked in Biocon for a year between 99 and 2000. So in a way, the forest was created by the seeds that fell off from Biocon, so I'm part of it. What we do is slightly different, but I want to set a little context. I put in a few slides so that I can leave this behind for some of you who may want to go through it. I think the century of biotech is kind of emerging now. And the next 100 years or so will not be dominated necessarily by AI and robotics and space, but most probably from biotech. And this is what I see and hear, because end of the day, we need food to eat, we need clothes to wear, and many more things for the 9 billion people who may be inhabiting this earth in the next few years. What's interesting is, while the equity markets divide the market into FMCG, pharmaceutical, biotech, etc., more than 60% of all industrial inputs are bio-derived. The bioeconomy is much larger than what the equities markets really assess, and it's far beyond just healthcare alone. I'm here to introduce what I would call the next frontier in biotech, which is not limited to human health, but for human way of living, for continuing to live the way we do with the kind of food we eat or the kind of preferences we have all over the world. In fact, this is a great time for biotech because this convergence of geopolitics, economics, and technology. AI is changing the way we do biofoundry, but at the end of the day, everything in biotech, whether it's CAR-T cells or insulin or anything else, has to be made using a physical infrastructure. So today's age of biotech is not limited to the work we do in our labs, but actually putting steel on the ground and creating this infrastructure. So in fact, my experience, I made several products, recombinant products for human health in the past. And I found that 70% of the translation happens in manufacturing and lesser so in the lab after the technology is developed. So what we do is we use the same technologies used to make insulin. In fact, we made trypsin for Biocon, with which they were able to enter into the regulated markets. It came from animals before. So over time, we found that there are areas that we could actually build scale and lead from the front rather than being a biosimilars company or another company following an innovator. We could actually make bioproducts which are not limited by regulations or patents. We could make products that mimic nature and actually create some really awesome offerings for the world. In fact, we are trying to build scale never imagined in India. If you collect the entire fermentation capacity in India, including all the leading companies, it's a fraction of what we think we should build in one site. When I built manufacturing plants in the past, 70% of my time was managing utilities. 20% was biotech and 10% was regulatory and QA/QC. This is the fact of life. If you went into Biocon, you'll see there are more people managing utilities, uninterrupted power supply, wastewater, and whatnot. In fact, the best utility companies are the guys who run sugar mills, distilleries, power plants, etc. So why not co-locate them? So that's our strategy. One of the things I want to bring to notice to many of you researchers here is there are opportunities, just too many of them. It's about what you want to chase. I'm going to skip this, but this is where I want to spend a little time to explain what we are doing here. A lot of science dies in the lab. They don't make it to unit economics, they don't make it to validation. There are many companies funded by BIRAC, DBT for scale up, for supporting startups, but once they start running their vaccines or their programs, they really don't have space for other startups to go move and test their products or validation. In fact, most scientists I know are struggling to move what they did in the lab to create real value through validation. I think that's what we're bringing to the table with THSTI, being a public funded organization. We're bringing our experience in both GMP and non-GMP manufacturing to fill this important valley of death. This is where most companies don't make it, most investors don't fund, most startups fail. Because even to run a single GMP batch for a validation process is like 2 to 3 million even in India. And they are competing with other global companies who want to hire services of, say, Syngene or anybody else who are there. So I think this is an unmet area, but it also needs somebody who can share its experience over here. So we as a company are not here to take something from THSTI, but we are here to come and give our expertise to build what I call the first of its kind lab to validation scale GMP and non-GMP capabilities. And I'm very proud to be here and associated with Dr. Karthik and others. I found some of the most supportive, forward-looking leaders in this institution. Thank you.
Thank you, sir. The floor is now open for questions, if any. Next, we invite Dr. Syed Khalid Ali from Panacea Biotech Limited.
Okay. Hello, good afternoon, everybody. And I would thank the organizers for allowing me to speak about the collaborative projects between Panacea Biotech and THSTI. And today I'll talk about 3 projects that are going to move into first in human or clinical phase. And that's why the title. I am Dr. Khalid. I am Chief Scientific Officer at Panacea Biotech. And Panacea Biotech is a 30-year-old fully integrated biopharmaceutical company. And this slide depicts the impact that we have created over past 30 years of our existence. We are one of the only 2 companies to have supplied more than 10 billion doses of polio vaccine and played a major role in eradication of polio from India and the rest of the world. We are the company which has developed fully liquid pentavalent and hexavalent vaccines. These are pediatric vaccines that every kid born needs to take. Includes DPT, Hib, hepatitis B, polio, and so on. And in addition to that, we have a GI, diabetes, organ transplant, and oncology franchise, which are treating tens of millions of patients. And recently we have forayed into food and nutrition, and maybe next time I'll talk about that. But coming back to the collaborative projects, we are working, today I'll talk about 3 projects. One is a COVID VOC, broadly protective COVID vaccine. We call it NanoCovac. We have completed preclinical studies with this, and it is heading towards phase 1. The second project is a broadly protective beta coronavirus vaccine project for pandemic preparedness. And the third project is Vaxipox. It's an Mpox vaccine. People may ask that in today, when COVID ceases to be a major public health problem, why are we working on COVID? But this news clipping is from yesterday. And so we do have COVID, and there is a possibility it may become a public health problem in recent future. And that's why we continue to be prepared for that. On the right-hand side, you have figures, global figures from WHO on the COVID vaccine, on the COVID disease burden. So with this vaccine candidate, we have completed preclinical POC, CMC development and toxicology study, and it is ready to head into phase 1 clinical study. The next project that I'm going to tell you about is a broadly protective beta coronavirus vaccine candidate designed to protect against SARS-CoV-1, CoV-2, and MERS. So single antigen protecting against 3 major coronaviruses. And this project is supported by Coalition for Epidemic Preparedness, CEPI. And this is a partnership with THSTI we are particularly proud of, because our scientific prowess is recognized by CEPI and the global experts, because we demonstrated preclinical proof of concept, and then our project went through an evaluation phase called stage gate review. And we passed through flying colors. We got feedback that the quality of our presentation and our data was better than everything else that they have heard. So this is a major achievement in terms of an industry-academia partnership. In this project as well, we have completed CMC development. We are preparing to start preclinical toxicology study. And you see a panel bottom right-hand side. Green line is our vaccine, and the other lines are other candidates. And as you can clearly see, we achieve 100% protection against MERS as compared to other vaccines and other candidates. And we had similar data for SARS and COVID as well. With that, I move to the final vaccine candidate, that is Vaxipox. It's a Mpox vaccine candidate. Here again, we have completed everything. We have initiated preclinical toxicology study. It is ongoing as we speak. Maybe in a couple of months, we'll start GMP manufacturing. And early next year, we plan to take this candidate into phase 1. Again, on your right-hand side, you see a panel. Green line is vaccinated animals. This is preclinical POC. So as you can see, vaccinated animals were protected against challenge from vaccinia virus as well as the wild type circulating Mpox strain as well. I'm only showing vaccinia data for the sake of time. And so to conclude, I think we have 3 projects that are moving into phase 1. And we expect Mpox to be the first one to go into phase 1 starting February, March next year. The other point is all 3 vaccine candidates are fully made in India, discovered at THSTI, developed at Panacea. And that's why we own all the intellectual property with regards to the antigen as well as the adjuvant and formulation. And yesterday there was significant discussion around industries contributing to development and R&D activities. So NanoCovac and Vaxipox are fully self-funded, and we have taken them to talks, and we are going to take them into phase 1 study. The third candidate, BPCV, as I mentioned, is funded by CEPI. So this is a brief update since I had all but 3 minutes. So thank you for your time and attention.
Thank you, sir. The floor is now open for questions. Yeah. Sir, please.
Yes, Dr. Ram.
Dr. Khalid, it was a nice presentation. So what is your strategy for going into first in human for all the three? How are you scouting? What is your plan?
So I think since these are novel vaccine candidates developed on a novel platform, we plan to do a dose escalating phase 1 study in adults to begin with, since adults are the healthiest of all the age cohorts. We will start with a dose which is much lower than what we have tested in preclinical immunogenicity and efficacy study, where we know the safety is ensured, because as you know, phase 1 study is the primary objective is safety, and then slowly age de-escalate 3 to 4 different doses, and ideally, we would like to hit the safety ceiling with our doses, but that there are resource constraints. But a dose where we are ensured a protective immune response and which is safe. So that is the point that we are trying to reach with these studies.
Thank you. I also invite you to visit our phase 1 unit. It has improved since you last visited.
Yes.
And it's open invitation for others as well.
Yes, yes. And that's where, that's at least one of the studies. Let's plan one of it there.
Thank you.
Thank you. Yes, Dr. Karthikeyan.
So, thank you. I'm just going to put you on the spot here.
Yes.
Because whole of Malaysia is here. So, see, this is a very good direction. This is one of the few times that Indian industry has taken up something indigenous and is going to take it to market,
Yes.
right? So, this means that there needs to be a fundamental shift in the way Indian industry thinks. Less biologics, less biosimilars, less all those things, and more
novel.
So, which means investment.
Yes.
So, I mean, it's a good case for you. So, what going forward, how much of your revenue will go into R&D, basic R&D?
So I think you should look to your right. And
I know
maybe
that. I know that.
That's where the real answer lies. But Dr. Jayan, I think, would be better.
Hi, this is Rajesh Jain. Thank you very much for inviting us on your foundation day, and thank you. You did a good job. Very
Thank
good.
you.
On an average, I think 5 to 6% of our revenues we invest in innovation, in R&D. And when we say R&D, we are saying majority of this money goes into risky projects, because you cannot say that these novel molecules will go to the market and one day commercially be successful. So this is what we have done right from the beginning, that we want to invest money in innovation. And it was not only the new molecules that we have invested money, also new types of formulations, drug delivery systems, even adjuvants we have worked upon, trying to develop an ecosystem through which not only Panacea but other collaborators who want to rise from India and try and create affordable innovation could also be supported through our efforts. So I think it's not just the money, but your ability to face failure continuously, one after the other, year that you are investing money in. That is what Panacea Biotech as a company is. And when your scientists are failing, when your experiments are failing, how do you actually inhabitate them, accept them into the company, and yet make them perform one day? I think that is the DNA that we have. It's not just money, sir. It's so much things put together. Thank you.
Thank you. No, it's a very good example that you've set for Indian industry. So I think, others can also take follow.
Yeah, I didn't mention this, but I think this would not have been possible without your support and support of Dr. Garg previously, I think, throughout. So it's not just technology transfer. We hit multiple roadblocks, some regulatory, some otherwise, and Dr. Karthikeyan and Dr. Garg were there to steamroll the problems and pave the way for us. So thank you.
Thank you, sir. Next, we invite Dr. Shiva Natarajan from Miltenny.
Good afternoon, everyone. It's an absolute honor and privilege to be here. And I can't thank enough Dr. Karthikeyan and his entire team. You need to be incredibly proud of what you've achieved, because the variety and velocity of what has happened between our first meeting and today has been nothing short of splendid. So I'm going to talk something different today. It's something that we envisioned possibly 2 years ago, but it has become a reality now. And that's a CAR-T manufacturing training that we did over here. And for those of you who do not know me, my name is Shiva Natarajan. I work for Miltenny. And I also am a part-time teacher in a management institute. So naturally, teaching and training is something that's very near to me. So something about Miltenny. The previous speaker alluded that biotechnology is almost 100 years old. And what is significant for us over here is that we are 35 years old in this, right? And which also means that we have a head start. This is of particular interest of those who are in research, because we somewhere compared the biotechnology industry to amoeba, because it keeps changing shape. And what we are seeing in the last few years has been tremendous movement in terms of development as compared to the previous 25 years. So therefore, as an organization, we do have a head start as a big advantage. The second thing that you see is a number that says 23, which means that 1/4 of our employees in Miltenny are scientists. And this is very unlike in any commercial organization, where the number is likely to be much lower. The vision possibly is because of our founder, Stefan Miltenny, and he comes to our office on a bicycle. And when somebody asked him why doesn't he have a Porsche, he said that if I had that money, I'll put it back on research. And that's the ethos and credibility with which we enter the biotechnology area. So our offices are located globally, but of particular interest are two areas, where one is in the US in Gettysburg, and the other one in Germany in Tetro, where we do have our offices and factories for the lentiviral vector. And recently we've opened our office in Hyderabad here as well. So these are our offices. So for those of you who are keen to visit our office, please do reach out to us. It will be a pleasure to host you there at our cell factory in Cologne. Now, to my topic, I wanted to speak about this a little more. And if you see that on one side, you'll see that the cost of developing a drug is close to 2.6 billion. It's but natural that most of the pharma industry, therefore, is shifting towards biotechnology. And you see the rapid growth of biotechnology that's happening over here now. So most of the multinationals that you see in the news now are shifting towards venturing into areas in biotechnology than in traditional pharma. And therefore, this is possibly the right place to be. I know I'm talking to brilliant minds in this area who understand CAR-T therapy really, really well, but the story of CAR-T is never complete without talking about the patient. So every time we look at trial results and we see a Kaplan-Meier curve, we know there are patient numbers behind that, but each number has an individual patient. And all of this started with one particular patient, which I want to highlight about today, and I'm sure most of you have heard about Emily Whitehead. So for those of you who do not know what CAR-T therapy is, it is what I call always as a rocket science of cell and gene therapy. You get patients' blood outside, re-engineer it, and then push it back, right? So it's not as simple as what I just mentioned, but that is what was offered to a patient called Emily Whitehead. When Emily Whitehead was enrolled in a trial, she just about had few weeks to live, and that's when her parents were introduced to something known as CAR-T therapy, and then they said that can we then therefore put your daughter on this trial. And one thing led to other, and the process that I just mentioned is what is mentioned over here, but you can see the rigor that is needed. It is not just about taking blood out and then pushing it re-engineered back. The rigor that goes behind this is different from just, for an example, just dropping a paracetamol. It doesn't work like that, because in this case you don't have a product. The process is the product when it comes to CAR-T. And therefore, Emily Whitehead was the first one who was just given a few weeks to live, who was given CAR-T. And of course, she proudly posted this image which said that she is 1 year cancer-free. And she goes on doing it every year now. And that is why CAR-T became such a big success globally and for us in India as well. Now, coming back to why we wanted to be passionate about what has happened in the space of CAR-T is because in our ethos and values, there is also a mission that we say that we need to train people and get them up to speed in terms of technology when it comes to cell and gene therapy. Now, if you see the total requirement as an ecosystem when it comes to CAR-T manufacturing, the GMP premise, of course, is the most important one that we start. And then, of course, the next one is the product that we have, the processes and the SOPs that are needed to manufacture CAR-T, the procedures and worksheets that you need to follow. And of course, all of this is incomplete if you don't have the final, most important pillar, which is the final P, which is people. Now, this is one part where, as I mentioned, we are very proud to tie up with THSTI and work on a program. So I'll give you a bit of glimpse on what it is all about. So CAR-T manufacturing, from an expertise perspective, we do have that within Miltony in terms of not just SOPs, but what needs to go into a regulatory framework, what kind of training is needed therefore, and offering an entire cell manufacturing enabling package which runs for at least 6 months to train somebody to get them up to speed on CAR-T manufacturing. And this is something that I'm very proud of, because 2 years ago when we started, I have to tell you that we have been invited to many organizations to partner with them, to collaborate with them, but this is one collaboration which did see the light of the day. So we tied up, we had an LOI with THSTI at that point of time, and honest confession from my side is I was clueless in terms of what I'm supposed to do and how I'll take it forward. But one thing led to other, and of course the team at THSTI has to take full credit for that in terms of paving the pathway for that. We tied up with DBT, and then we launched India's first CAR-T training program. India's first CAR-T training program, and that is something that all of us should be really, really proud of. This is something that is just a beginning that we had, but just to give you a glimpse of what we did was, we were also in the learning mode and we wanted to understand how do we make this a success, so we were testing waters over there. The best part about this was that for a personal attention, we said that we can't do more than 15 in a batch. Until the last minute, we were getting requests to include people into that batch, and then we finally agreed to a number of 20. And we gave a time of around 2 months for the registrations to fill in, but the training program ended up getting filled up in just about 15 to 20 days. So we brought in our first prodigy. I think Dr. Lakshmi alluded about the MRC over here and the prodigy that we have over here. And this is something that we installed before the GMP facility at THSTI was ready, and then we said that we will possibly use this for training. This is a batch just to give you a glimpse of our people flew in from Germany, and of course we had our colleagues over here in India who were certified trainers who gave them good training. I'm leaving you to read the comments over here in terms of actual verbatim quotes from some of the participants who were thrilled about the participation and what they underwent for the training program that was conducted over here. Now, as much as I spoke about what we have done in training, I just want to also mention that this is the tip of the iceberg when it comes to CAR-T manufacturing. You can see that there is so much that we need to do. The training program sometimes even runs up to 18 months. We are just about to start a validation run in one of our sites, and there are around 81 SOPs that my team is working on just for conducting 3 runs. That's a rigor that we have. So just as a food for thought that it is just a beginning, but there's a long way to go in terms of equipping yourself on CAR-T manufacturing. These are numbers that speak for themselves. Now, as much as I've spoken about what is CAR-T, why is it that it is of so much of interest for people in India? It's been really a while that we saw something like this appearing in one of our national dailies. And what you see here as a comment is actually from Times of India, and it also had a recognition by the highest dignitaries from the country on what we've done in CAR-T manufacturing. That's on one side. The other side is something that you should be super proud of, because for the 25 years that I've been in the industry, hardly have we seen any Indian brands featuring in the multinational landscape. And among the plethora of companies that you see on the other side, there are 2 companies from India that are mentioned as CAR-T manufacturers. One is, of course, Immunil with Amit leading over here, and the other one, ImmunoAct. So we should be super proud of what we've done in the CAR-T space. And when we started off, we just had about 3 CAR-T companies in the country, but now we have almost around 9 of them working. And the rapid pace with which this is moving is something that we should be super proud of. As much as I've told about all good things that we've done and what is happening in the CAR-T space, I also want you to leave with this infamous message that I saw in one of the congresses where I clicked the picture. It talks about what is known as a car traffic jam. And if you see the highlighted part over here, it talks about an entire ecosystem. It doesn't just talk about people, it talks about processes, it talks about the requirements, the regulatory approvals, and everything else. So in order to make CAR-T a successful initiative, I would leave you with a thought that if there is good collaboration, then it is a brilliant way forward. And my concluding message over here is that I know I'm talking about CAR-T to a group of individuals over here who are brilliant and individually super powered in their brain. We also have a plethora of experience who's sitting in the front seats over here, cumulative of more than around 100 years of experience. So the value of a network is directly proportional to the number of people who are connected to the network. So I will encourage you to speak to as many people as possible to pick their minds in terms of how do you want to make the journey more successful. Right? So that's my message to all of you, and thank you so much for your attention.
Thank you, sir. The floor is now open for questions.
Go easy on questions. I stopped going to school because of that. So.
Yeah, sir, India has become global leader for the reverse engineering and biosimilars, but still India has become global leader for the reverse engineering and biosimilars, but still we lag behind early phase clinical research despite we have good preclinical evidence. Why is happening, and what are the challenges as you successfully manufacture CAR-T, you must experience what are the challenges we are facing in scientific and regulatory in India. And another thing I want to know, as a clinical trial research student, what are the what are the similarities that we can enter and bridging the gap between two?
First of all, brilliant question, and thanks for that. When it comes to our role in trial, right? So we were famously called as the pharmacy of the world because we were making generics, and that's I'm using this word very carefully. That's unfortunately also become a perception builder for us to say that we make products that are super economical in India, and in effect, it also led to another problem in the ecosystem, which was brain drain. So people started leaving the country for want of better research facilities outside the country, and then they were hesitant to come back. This situation has rapidly changed over the last 5 years. Now, India is seen as a destination for us to do trials. We have started doing our first point-of-care trial in India, and there are many companies which are looking at India as a strong foothold for that. So what was happening earlier clearly is changing at this point of time. That's one. Second is, to your question, the answer is the very reason that you're in this room and you're asking this question itself is a big development. 10 years ago, I wouldn't have imagined a question of this kind coming. Right now, we have a facility which is now ready for conducting clinical trials. And this wouldn't have been possible without a network of people coming together. And right now, we are in that crucible. But have we really achieved that step to take off? No. We are still building the runway and we are trying to get the planes in order, that's it. But once we take off, I think then I would possibly foresee a lot more velocity in terms of uptake when it comes to clinical research. But the beginning is good. The future is clearly behind us, and whatever is in front of us is surely bright. Okay? Yeah.
Good afternoon, sir. Sir, actually CAR-T cell therapy is a very expensive therapy. So I wanted to ask, how do you plan to make it affordable for the rural population of this country?
Okay. I get this question very often. So I would encourage you to read a trial paper from CMC Vellore on point-of-care manufacturing. The outcome of that trial was not just the clinical data that came out of the trial, but it also showed that you can do low-cost manufacturing, and the price of CAR-T that we've done in India is 1/10 of what you get globally. So that is just the beginning. Once you get unit economics kicking in, which is a little too early at this point of time, then things will be much better in future. And the second thing is something that I'm passionate about when somebody talks to me about expensive drugs. I had a mentor who asked me a question at one point of time asking, do you know what is the most expensive drug in the world? And the answer to that he gave took me a while to understand because it is very philosophical, and I will repeat that to you. He said that the most expensive drug in the world is a drug that doesn't work. Right? And our entire effort is to make sure that you have a product that is really good. Otherwise it becomes very expensive, even if it is costing a few thousands.
Okay, thank you so much, sir.
I had a quick question.
Yeah, sorry.
Excellent initiative about the training and capacity building. I just wanted to understand in your next round, I believe you are going to have one, how much involvement are you going to have from the public sector, medical colleges and the institutions, because, there's a lot of disease there. So would there be an initiative in that direction?
Absolutely, madam. Thank you again for this question. So the training program that we started off had a mix of good representation from both private as well as from public. And it is agnostic of anyone. They just need to be basically trained to understand what is in cell and gene therapy and a flair for manufacturing and understanding of CAR-T therapy. So naturally it is open. And of course, we never take for granted that some of the best minds are in the public sector, and it is always open to that. The limitation that we have, however, is what I mentioned. So the number of people in each batch has to be kept at a low because of the requirement for the tubing sets, the reagents, and the personal attention that is given to them. So if the frequency of the training program goes up, I guess this problem can get addressed to a large extent. But we just kind of started rolling up our sleeves and we started working, but a long way to go, but we will certainly get over there. But the answer to your question is, they'll be, of course, welcome to the red carpet.
Hello, this is Dr. Priyanka from Panacea. So I have a question. Since long we have been hearing that Milteney, the system which has been very accurate and reproducible for making the CAR-T. But in this space, are you looking for any kind of open system? Because Milteney is a very closed system. So if you would like to make it cost effective, okay, and to reach the rural area. So what are your thoughts in this?
Okay. So in fact, the example that I gave, the first example was an open system when it came to CAR-T. And each open system and each handling that you do on the CAR-T sample will of course require that many extra SOPs and process and procedures to be done. People need to be trained, etc. So the cost actually gets built up when you do an open process, and which is why we came to the technology of a closed process at this point of time. So ours is the first fully automated closed CAR-T manufacturing, which takes care of the disadvantages that currently exist or existed in the open process. So we will hesitate, of course, going back to the open system, and we'll be focusing more on the closed system now. That's the vision.
Thank you.
There are no more questions. Thank you so much. It was a pleasure addressing all of you. Thank you.
Thank you, sir. Next, we invite Dr. Gitanjali from Medanta Groups. And Dr. Sanjay Mittal.
Good afternoon to you all. Well, I'm proud of myself being here on behalf of Medanta, the Medicity, to partner with THSTI for the health and development of science to save lives. This has been the bottleneck of all the previous speakers who said we are doing work here to save human lives. I bring down hello from Dr. Trehan, who has this vision of Har Jaan Ek Anmol. Every life is valuable, and that's how we had laid down the foundation of the Medanta the Medicity. Proud of being the ranked first hospital in India among the world's best news week 7 years on the continuum. We are having advanced therapeutic capabilities, trusted destination of the complex cases, and we have a beautiful infrastructure, we all know. But this is of no good if we are not able to place good therapeutics to our patients. So our deliverance actually depends upon our patient population, our patients' plights, and what we can give to those patients to save them and to make them live better. We have, as for now, 6 hospitals, almost compounding to 3,000 plus beds, 2,000 plus doctors, and one integrated model of bringing together clinical care, research, education, and innovation. We have been a part of a lot of new technologies which have been introduced to help our patients recover faster, and also a huge investment into research portfolio. We are dealing with clinical research, and the foundation stone for clinical research is ethics, patient safety, and quality. And this is the reason why we are looking forward to better therapeutics. We are into clinical research from bench to bedside, but we are not having right now the capacities to build the products from the good manufacturing standpoints. And that is why this handshake is so very important. The concept today, as much as I know about medicine and cardiology, I'm a cardiologist though, is that the knowledge today is not even a drop in the ocean. And we all have to work together to build that knowledge for our safety, our patients' safety, and humans' development of science. And this is the reason we actually, from our physician side, have 2 ends of the development cycle. We have the problem, or the demand, I say, and we have the deliverance, or supply. Because the problem eliminates from our failures to supply right treatment, and the patients in front of us actually suffering because of want of treatment. And we have new therapies which we are there to supply to the patients who actually deserve them. But there is bridging, which the THSTI probably would help us best, is the development process, the translation in science from the bench to the bedside. I think the continuum is so very important that there may be a demand, but if there is no development, or the development without a demand is useless, and development without a supply is useless too. I was hearing a beautiful lecture from the previous speaker, but I say a costly medicine which is not affordable to the patients or the people is just like showing bread to a starving man and saying that I am going to give you this bread only if you pay me something. So it's a bigger crime in my mind to provide a cost ineffective medicine to the people. And that is why developing these therapeutics in our country at an affordable cost is so very important for the whole community. We are going to have a collaboration with THSTI on these 4 points: joint collaborative research across basics, translation, and clinical sciences, capacity building. We have manpower. We are ready to invest into the manpower and the problem-oriented science development to the THSTI. Thank you. Thank you very much.
I'll just add a word here. I would like to thank THSTI for reaching out to Medanta to collaborate, for initiating this collaboration between the 2 institutes. As you all know that Medanta has a huge patient base, and THSTI is working upon new technologies, new products, and this gap, this bridging of this gap between the hospital and the actual academic institute, I think this is where we are stationed. And it is very important that more institutes like these can collaborate with each other so that the first-in-man studies, where we are highly focused on, can be initiated and can be done in a very cost-efficient and a timely manner. So that is the bottom line of this collaboration that we have here, and we really look forward to that. Thank you. And any questions for Medanta?
Thank you. Thank you. Next, we invite Dr. Sanjeev Kumar from Amrita Hospital.
A very good afternoon to all of you, and a real honor and a privilege to be here in such a fantastic institute. We visited this institute a couple of weeks back and didn't realize that it would translate it so very soon. I'm Dr. Sanjeev Singh. I'm Medical Director at Amrita Institute, and we are neighbors with THSTI. We are at Faridabad, which is a 2,600 bed facility. Other than the clinical work, there is a medical school, nursing school, allied health science PhD program. We used to say we have a large research block. After visiting THSTI, we say that we have a decent research block at our facility. We also have a large simulation academy because we realized that when we were graduating, there were 250, 300 colleges. Now there are 818 medical colleges, and they all need to be skilled and trained. While getting the facility ready, we also realized that we need to marry with the technology, and there's only one engineering college which we have opened at our facility, though it's a health facility, which is AI in medicine and data sciences. And we are making it mandatory for all the UGs, PGs and the faculty to be AI and chatbot trained, and that's the future, to make them future ready. During this course, we also realized that art and science of medicine, science is fantastic, and you just heard Medanta and so many brilliant people. Art of medicine practice is diluting, and so we have opened a school of social and behavioral sciences, and we now think that it's a complete campus with humanities, with technology, and with clinical skills. And while during our visit, we realized that there are many common areas of interest, would be maternal and child health, antimicrobial resistance, infectious diseases, early biomarkers, and our facility and our clinical faculty are available for this particular, and they're very excited for this collaboration. We would really like to thank THSTI for opening this gates for Amrita, and we look forward for this an extensive collaboration which would ultimately be of large benefit to the society. Thank you so much.
Thank you, sir. Next, we invite Ms. Devhuti Palan from Arundhati Healthcare Private Limited.
Good afternoon, everyone. I'll jump to the product that we are developing and then tell more about me and the company. We are developing Clever Cup. It's a platform for clean urine collection. These are my team members, and I'm blessed with mentors to fill in the gaps. We are at an early stage, a startup, so we are yet to build the institute, and we are very inspired by all the previous speakers. And the problem that we are trying to solve is the contamination in urine samples. So we have tried to incorporate the verbal instruction when we conduct a trial or a study, that patients will give the clean midstream sample. But in real life setting, our paramedic staff is not able to address this gap. We cannot instruct each and every patient that comes to the lab that these are the instructions that you have to follow and then give a clean midstream sample. So this is the gap that we are going to solve with our product called Clever Cup, which has 4 compartments, and it has been developed to automatically segregate urine in real time in first, mid, and late to ensure there is no contamination from skin microflora or perineal microflora, and we are able to find out the actual culprit behind the infection. We have patented it, and today the assignment will be done with the presence of Honorable Minister, sir. The unique value proposition for our product is how we cater to primary healthcare centers differently with a device that captures the entire urine stream. So we are not at all dependent on restrooms. It works with privacy curtains. And for the urban setting at the tier 3 cities, it has exit valve for the toilet. So the product has 2 categories in broad terms, that it works without restroom for primary healthcare centers, and it works with restrooms for the hospitals and diagnostic labs. When you look at the market opportunity, daily 36 lakh units are used across India, and that's a conservative calculation with 60 tests per hospital. So if you are able to capture 10% of the market, still we can build a sustainable business out of it. Our current goal is to lock in the design and go for pilot manufacturing and investigation of the product at the hospital site. When you look at the other products in the market, nobody has tried segregation like we have done with different compartments to segregate in first, mid, and late. And this is the product development journey. We started conceptualizing it during our biodesign program. So I've been here in THSTI from past 3 years, and I am very grateful for the opportunity that during the clinical immersion, we found out this unmet clinical need, and then we took it forward. Now we will commercialize the product through the startup. This is some insight on the benchtop testing that we have done for the device. And I'm grateful for the whole ecosystem to encourage me with fund and grants to take this product forward. And this is the broad business model canvas for us, that right now, key partners are THSTI, RCB, Venture Center Pune, IKP Knowledge Park, FITT IIT Delhi, BFI and M Pragati, NSR Cell IIM Bangalore, Ravi Plastic Private Limited, Quick Shape Private Limited. For a startup, it is very important to have right partners and to be placed in the ecosystem in such a way that we can thrive. And I'm glad that I got this opportunity here, from the inauguration of the biodesign program to the expert feedback from Shinjini ma'am, Kang ma'am, and then with our ED sir to take this product forward. And now we are at a stage where we will get the assignment and we'll take this product ahead. Thank you.
Thanks to all the distinguished guests. I now invite Dr. Nitya Vadva, Senior Professor, THSTI, to welcome our esteemed guests.
A very warm good afternoon to everyone. It is my privilege to welcome you all on the 17th Foundation Day of BRIC Translational Health Science and Technology Institute. We are delighted to welcome Professor Roger Fu, whose Foundation Day lecture earlier today offered inspiring insights into cardiometabolic research. I also welcome our collaborators from academia, government, industry, and funding agencies, and every member of the THSTI family, our faculty, scientists, students, and staff whose dedication drives the Institute's achievements. As we gather today, we remember with deep respect Professor M.K. Bhan, whose extraordinary vision gave birth to THSTI. His belief that science must ultimately serve society continues to guide everything that we do. Every partnership we build, every innovation we translate, and every life we seek to impact is a tribute to that vision. Yesterday, Syngin 2026 brought together over 300 participants from academia, industry, government, regulatory agencies, and funding organizations. The energy, dialogue, and new connections forged during the meeting exemplified the collaborative spirit that underpins innovation. It is particularly fitting that today's Foundation Day celebrations will also witness the formalization of new partnerships with industry, carrying forward that spirit of collaboration. As THSTI enters its 18th year, we remain committed to scientific excellence, meaningful partnerships, and translating discoveries into better health for our people, contributing to the vision of an innovation-driven and Atmanirbhar India. On behalf of the entire THSTI family, I sincerely thank you all for joining us today. We hope this celebration honors our journey, inspires new collaborations, and reaffirms our shared commitment to advancing human health through science. Thank you, and I wish you all a memorable Foundation Day. Thank you.
हमारे लिए अत्यंत हर्ष और गौरव का विषय है कि हमारे बीच भारत सरकार के जैव प्रौद्योगिकी विभाग के सचिव एवं ब्रिक के महानिदेशक माननीय डॉक्टर राजेश एस गोखले जी ऑनलाइन उपस्थित हैं। मैं आदरपूर्वक डॉक्टर गोखले जी से अनुरोध करता हूं कि वह अपने प्रेरणादायक विचार हम सभी के साथ साझा करें।
Namaskar to all of you. THSTI, distinguished industry leaders, researchers, students, and members of the THSTI family. I'm getting it. Sorry, I'm over there. I've joined.
No.
Honorable sir, we can hear you.
I think I go back. What is the problem?
So Karthik, can you hear me now?
We can hear you now.
Okay, I think fine. I think for some reason,
You actually share another window from there, and that is causing this problem right now because it kind of gives me a second window, and that is
You're audible now. You're clearly audible now. You can go ahead.
okay, right.
Yeah,
No, but I think
when you're replicating my window, I'm getting an echo back again, actually. So now this may be okay. Right now, maybe you've switched off your mic. Okay. All right. Thank you very much. First of all, my apologies. Let me start with saying apologies that I couldn't reach there. We had an event actually with Honorable Minister, and Minister was late due to the cabinet meeting, and therefore the entire schedule got a little bit haywired. And I think if he's also not able to make it there, since he has a follow-up meeting with Finance Minister today. So, but I thought I should join you from here, and I think it's such an important day and such an important event actually that's happening there right now. It's quite remarkable what THSTI has been able to do over the years. So I thought I should come and say hello to all of you. So it's a real pleasure to join you today as we celebrate the Foundation Day of THSTI. This is not just an opportunity to look back what we have done, but also to really plan for the future of our country, future of our institution, and future of BRIC. And therefore, I think this has all been possible because of the hard work that has been put by all the people of THSTI family. And I would like to congratulate the entire THSTI family for its leadership, its scientific community, researchers, technical teams, administrative staff, students, and also, collaborators, as well as the industry leaders who are there actually. I think it's the people who build these institutions and organizations, and we are only part of your perseverance and your belief that science can make a real difference is what is really uniting all of us together. And thank you very much for all of you coming and participating in this big numbers. I think when THSTI was built, it was built with a simple thought process. And the whole thought process was, can we develop an institution that can have a science to translation conversion eased out by a mechanism that is which can be institutionalized through a process. I think it was always a wonderful dream, even though it's a simple idea, but it's a very powerful thought process actually. And I think today we are at the 17th Foundation Day. All of us are very happy to see this buzz around the THSTI activity, which is really gratifying to see. The journey from laboratory should not end in publication only, and it should become some part of either it should be developed as a vaccine, or it should developed as a diagnostic, or should develop into therapies. And ultimately, a better health outcomes will emerge out of it actually. So I personally believe that this was the vision that Dr. M.K. Bhan actually started this organization with, and I had the pleasure of working with him during the initial years of this THSTI setup, and I still remember those early meetings actually while he was guiding this institution around. I think it is now gratifying to see that THSTI has earned recognition not only within India but across the world as well. For example, WHO Regional Training Center for Biomanufacturing, Global CEPI Network, all speaks of large amount of amazing facilities that have been set up, talks about its credibility and capability in terms of what it has been able to do in a very short period of time. In fact, for an institution, 17 years is a very, very short period of time. I mean, typically the institutions mature in the second decade of their organization, which is around post 20 years. I am also very happy to see that THSTI has never lost the sight of its purpose, and scientific excellence has always been given priority in every aspect of endeavor, whether it's to translate knowledge or whether it's to find solutions and improve the lives of people actually. I'm very happy to see that THSTI now is also nurturing innovation. In fact, the startups of various biodesign, school of biodesign people actually is very gratifying to see. And I think there are, if I'm not mistaken, 5 of you have initiated now companies over there and building enterprise, which will create not only good ecosystem, but also provide jobs for others to look at and work. And eventually they can become a global talent pool as well as a biotech leader in those industries perspective. I mean, clearly, as all of you would recollect, no institution can achieve such successes in isolation. It's not very easy, that institutions on their own can do it. The challenge, therefore, before us is fairly complex, and this demands collaborations across industry players, scientists, academia, clinicians. And in this context, the yesterday's Sinchan meeting was, I heard, was remarkable there. In fact, Dr. Dhananjay Tiwari was there from DBT to represent, and he told me there was a lot of wonderful vibrations around there. And I think that's been a yearly platform in which is bringing all the folks together. And I think this is a wonderful mechanism to build partnerships, and that can evolve into eventually into products for the people, for the best of the people. Also, from the government point of view, what government is doing is actually bringing together conglomeration of research institutions as a single organization is helping us to build partnerships, not only across within institutions, but across institutions as well, actually. And I must thank THSTI for becoming an active member of Biotechnology Research Innovation Council as well. I also wish to say that the various partnerships that have initiated, for example, the one with Panisha Biotech on vaccine collaboration with taking various vaccine candidates, and I hope they move faster than what it has happened in the last year. But I really look forward to, how to really take forward these various programs to the next level in the years to come by. What is also good to see is that THSTI is also investing into regulatory capabilities and providing specialized training and emerging technologies, which are building new generation of scientists and also, developing India's biotech ecosystem. In fact, one of the first biofoundry that is going to be set up there, which is for various areas of antibodies, monoclonal antibody development, as well as for microbiome work there, actually, which is again two very strong areas of research activities in THSTI. I must say, as we strengthen our scientific capabilities, we should also strengthen our preparedness for future. And that's where the MRC, which is Medical Research Center, and the upcoming platform for facilitation of activities is going to provide us new opportunity for collaborations and innovation, while reinforcing the capacity to respond to health challenges of future of the goals. I also congratulate all the MOUs and LOIs, letter of intents, that have been signed with industry people. I've been told that there are 16 of them that are being signed. And I think this is a tremendous milestone in the progress of our institution progress. And I'm very proud as a Director General of BRIC to say that THSTI is doing it in a remarkable way, actually. I must add that, Department of Biotechnology is fully committed to supporting THSTI and its journey ahead. And I'm very confident that institutions will continue to push the frontiers of science, forge stronger partnerships, and contribute to the building of healthier, more resilient, and innovation-driven Viksit Bharat. As we celebrate the Foundation Day, let's renew our partnerships, let's renew our shared commitment to scientific excellence, and also the innovation for the service of the society, actually. As you can imagine, biotech sector was very nascent just about 5-7 years back, actually. And as our honorable minister, who always keeps saying that the COVID gave the right momentum to it, actually. And really the vaccine platforms and the manufacturing capabilities that we have set up gives us as a puts us as a trusted partner of the ecosystem that can provide manufacturing across all the countries in a way that is not just strengthen our country's resolve, but also the world's resolve of how to take forward these innovative technologies, actually. And I very much look forward to how these institutions' capacities can be utilized by various stakeholders as we plan for bigger plans for making various research parks and science parks all across the country, along with biofoundries and biomanufacturing hub, to really inculcate a culture of biotech ecosystem that will be, completely transformative. Either it's got to do with human health, or got to do with material world, or got to do with energy crisis or energy issues, and various challenges that our climate change related program that we are facing together as part of this country's progress. I truly believe that India is at the threshold of transformation in terms of biotech ecosystem, not just from an R&D perspective, but from the innovation ecosystem that has grown because of the BioRack presence. So such a large number of startups have been created. At the same time, through biomanufacturing and biofoundry and the BioE3 policy, the government is investing into capex such that various innovators can use these facilities all across the country and take benefit out of without having to scale up their technologies, without investing in the capex technology, so that you kind of bridge the, take away the early investment that is really keeps you to move forward to actually. And I very strongly urge each and every innovator to really connect with this network of biofoundries in the country and take the best advantage of it. I once again warmly congratulate each and every one who is present there, all the THSTI family, the BRIC large family people, as well as DBT ecosystem, who is providing a tremendous umbrella of activities. And we hope all the very best for THSTI. And I think really proud of all of you for what you are doing. Let's grow from strength to strength and make this the enterprise of our country for the future of the world and for the best of humankind. Thank you again, all of you, and namaskar to all of you.
Thank you so much, sir, for your inspiring words, for your kind attention. For your kind attention. Honorable Minister is unable to join us today as he's occupied with his prior commitments. Moving forward, I would like to invite our distinguished guest, Professor Roger Fu, Professor Shinjini Bhatnagar, our Executive Director, Professor Ganesan Karthikeyan, and our Dean, Professor Jayanta Bhattacharya, to please take seat on the stage. Thank you. I dearly request our distinguished guest, please commemorate this event with lamp lighting event. Thank you all. We host an annual Syngin industry meet to build synergy in science through collaborations. This year, we continued the momentum through Syngin 2026, hosted yesterday to build partnerships that fosters academia industry collaborations. We will now announce the names of our recent partners and request the representatives to join us on the stage for the exchange of agreements. From Central Drug Standard Control Organization, we invite Mr. Dilip Rajput, Director, Administration, Drugs from THSTI, Dr. Nitya Vadbha, and Dr. Neha Chawla. From THSTI, we invite Dr. Jayanta Bhattacharya, Dr. Nitya Vadhva, and Dr. Neha Chawla to receive the agreement with the World Health Organization, Geneva, for the designation of THSTI as the WHO Regional Biomanufacturing Training Center for the Southeast Asia region. From Immunal Therapeutics Private Limited, we invite Mr. Amit Mukim, Chief Executive Officer, and Dr. Lakshmikant Satyagat Nikonta, Chief Scientific Officer, and from THSTI, Dr. Amita Avasthi. From Syngin International Limited, we invite Dr. Mrinal Kamili, Head Translational and Clinical Research, and from THSTI, Dr. Nitya Vadhva. Now we invite Dr. Sweetie Samal and Dr. Ramrachandan, Associate Professors of THSTI, to receive the agreement with Infecta, a Netherlands-based research organization for collaboration on controlled human infection studies. From Cipla Limited, we invite Mr. Vipul Gupta, Head Corporate Affairs, and from THSTI, Dr. Jayanta Bhattacharya and Dr. Nitya Vadhva. Limited, we invite Dr. Anuj Goyal, Chief Development Officer, and Dr. Anil K, Head Medical Affairs, Clinical Research and National Regulatory, and from THSTI, Dr. Nitya Vadva.
Bio Private Limited, we invite Mr. Subramani Ramachandrappa, Director, and from THSTI, Dr. Bhabatosh Das. Brahmin Botanicals Private Limited, we invite Mr. Samarth Singh, Chief Executive Officer and Managing Director, and from THSTI, Dr. Bhabatosh Das. We invite Dr. Jayanta Bhattacharya, Dean THSTI, to receive the agreement with Oxford University, UK, for collaboration on clinical assays. We invite Ms. Devruti Palan, former SIB Fellow, Founder and CEO of Arundhati Healthcare Private Limited, and from THSTI, Dr. Neeraj Kumar and Dr. Soma Patnaik. Invite Dr. Jayanti Kumari Rana, former SIB Fellow, Managing Director of Astris Innovus Private Limited, and from THSTI, Dr. Sushmita Chaudhary, Associate Professor, THSTI. From the Medanta Group, we invite Dr. Sanjay Mittal, Vice Chairman, Clinical Research and Clinical Cardiology, Medanta, Dr. Gitanjali Uppal from the Department of Research and Clinical Studies, and from THSTI, Dr. Nitya Vadva. From Biodesign Innovation Labs Private Limited, we invite Dr. Gautam Pasupuleti, CEO and Managing Director, and from THSTI, Dr. Amit Yadav. From Prolivent India. From Prolivent India Private Limited, we invite Mr. Prateek Shah, Head Finance and Strategy, and from THSTI, Dr. Ruchi Tandon. From Amrita Institute of Medical Sciences and Research Center, Faridabad, we invite Dr. Rajiv Kumar Singh, Medical Director, and from THSTI, Dr. Nitya Wadhwa. We now announce those partnerships that have reached significant progress from the time of signing the letter of intents. From Panacea Biotech Limited, we invite Dr. Rajesh Jain, Chairman and Managing Director, Mr. Harshit Jain, Director, Dr. Amulya Panda, Associate Director, Dr. Syed Khalid Ali, Chief Scientific Officer, and from THSTI, Dr. Amit Awasthi, Senior Professor, Dr. Sweetie Samal, Associate Professor, and Dr. Shabir Ahmed, former scientist. It is heartening to share with you all the progress of our collaboration on the development of broadly protective beta coronavirus vaccine candidate. From our initial engagement, it has progressed towards successful completion of the preclinical studies and passed the stage gate review by SEPI. The candidate will now move towards regulatory approvals for initiating clinical trials. From Miltenyi Biotech, we invite Dr. Sivakumar Natarajan, Commercial and Business Development, Mr. Vimal Kumar Pal, Director for Regulatory and Government Affairs, and from THSTI, Dr. Amit Awasthi, for continuing our collaborations after successful completion of first training program on CAR-T using Miltenyi's proprietary equipments. From Doto Health Private Limited, we invite Dr. Shama Kothari, Director, Partnerships and Fundraising, and from THSTI, Dr. Ramachandran T. and Dr. Kondinya Desiraju for the progress towards an agreement on for design and development of an AI-powered pregnancy care platform and portable high-risk pregnancy triaging tools. From Cure AI, we invite Dr. Shibu Vijayan, Chief Medical Officer, and from THSTI, Dr. Ramachandran T. and Dr. Kondinya Desiraju for the progress towards an agreement on design and development of an AI-powered pregnancy ultrasound reporting system with accurate fetal assessment tools. We now move to a very special segment of today's celebration, the prize distribution ceremony. Achievements and dedication form the backbone of any great institution, and today we celebrate a few of many who have contributed immensely. I kindly request our Executive Director and Professor Roger Fu to kindly present the awards to our winners. The award for a faculty member for the best published paper goes to Dr. Bhabatosh Das. We now have the award for a PhD student for the best published paper. The award goes to Mr. Jishnu Shankar. The next award is for a PhD student for the 5-year all-around performance. The award goes to Mr. Sunil Guchar and Miss Taruna Ahrodia. Moving ahead, we have the award for a faculty member for the overall contribution to institutional development during the previous financial year. The award goes to Professor Nishit Agarwal, Dr. Krishna Mohan Atmakuri, Dr. Gaurav Batra. The next recognition is for the administrative staff member for overall contribution to institutional development during the previous financial year. The award goes to Dr. Soma Patnaik and Mr. Piyush Gupta. Next is award for a technical staff member for overall contribution to institutional development during the previous financial year. The award goes to Mr. Narendra Kumar, Mr. Sudhendra Yadav and Mr. Shubham Gupta. Next is award for the best poster presentation. The first prize goes to Bharti Kandial. The second prize goes to Sunil Gujjar. The third prize goes to Saloni Sengar and Imran Ahmed. Next is the award for the best colloquium presentations. The awards go to Linus Augustin, Kajal Kamboj, Jyotsna Dandotia, Sneh Bajpai and Juhi. I now invite Dr. Amit Avasthi, Senior Professor, THSTI, to kindly deliver the vote of thanks.
Good evening, everyone. On the behalf of Translational Health Science and Technology Institute, it is my privilege to propose the vote of thanks as we conclude the celebration of the 17th Foundation Day. We express our sincere thanks to Dr. Rajesh Gokhale, Secretary, Department of Biotechnology, for his constant support, visionary guidance, and invaluable advice that continue to steer THSTI towards excellence. I take this opportunity to thank Dr. Dhananjay Tiwari, MD, BIREC, and Dr. Sajuddin, the Nodal Officer of THSTI, for their constant support and guiding us. Our deep appreciation goes out to all industry delegates, funders, policymakers who joined us today. Your presence and partnerships are vital to the success of both Syngin and the Foundation Day. We are equally grateful to the distinguished dignitaries from DVT, directors of sister institutions, and all our esteemed guests who travel from near and far to celebrate this milestone with us. Finally, an event of this scale is impossible without a dedicated teamwork. I would like to take this opportunity to specifically thank all the organizing committee members of the THSTI administrative staff and the IT team for their meritorious planning and the unwavering effort behind the scene to ensure seamless execution of today's program. Thank you. Thank you everyone once again for making this day a memorable success. Have a wonderful evening ahead. Thank you very much.